and welcome to Miss Estruch Biology, and in this video I'm going through all of Year 12 or AS Cambridge International Biology. So this is going to be a mega video. Get yourself comfy, you might even want to watch it at ×2 speed if you're a bit short of time, but this is all the theory straight from the spec that you need to help you to get the grades that you're aiming for your A-level. And if you do need any more help, then don't forget to check out the link in the description, which has got the link to my A-level notes, which covers all of the theory, so you don't need to make your own notes. And it includes the key marking points, key terms, and special examiner's tips for every single topic. But for now, let's get into this entire Year 12 AS video. So let's jump into then all the information you need to know for Cambridge International A-level for topics 1 to 4. And we're going to be going through 1 to 4, which is cell structure, biological molecules, enzymes, and cell membranes and transport. Start with cell structure, which is split into these 2 subsections. The microscope in the cell studies, and cells are the basic units of living organisms. So we're starting with microscopes. And there are 3 types of microscopes that you need to be aware of. Light microscopes, and then there are 2 electron microscopes. Light microscopes have a poor resolution, or at least it's poorer in comparison to an electron microscope, and that is because the resolution is determined by the wavelength of the light. And wavelength of visible light is longer than the wavelength of an electron. An advantage of light microscopes compared to the electron microscopes, though, is you can view living samples, which isn't possible with electron microscopes. And also you do get colour images without having to artificially add in colour to a photo afterwards. So then if we have a look at the first type of electron microscope, electron microscopes do have higher magnification and higher resolution, and that's because the electrons which are used to create the image have a shorter wavelength compared to visible light. The way the image is created is the electrons pass through the specimen. That's why it's called transmission, because the electrons are transmitting through the specimen, and they'll absorb different amounts of electrons, and that's how you get the different shades of grey to create the image. So a transmission electron microscope will enable you to see the internal structures of organelles. Scanning electron microscopes also have a higher magnification and resolution because they are creating these images with electrons which have a shorter wavelength compared to visible light. But this time you get a 3D image which shows you the contours on the outside. And that's because the electrons will bounce off the surface, and the way that they are reflecting is what creates that 3D image. So we've talked about resolution and magnification a little bit, but we haven't actually defined what they mean yet. So resolution is the minimum distance between 2 objects in which they can still be viewed as separate. So that is your key definition to learn. You could turn that into a flashcard. And with optical microscopes, that resolution is determined by the length of the wavelength of light, and as we said, electron microscopes is determined by the wavelength of an electron, which is shorter than for visible light, which is why electron microscopes have a higher resolution. The term magnification refers to how many times larger the image that you're viewing is compared to the actual object, and that's a formula that we'll be going through shortly as well. So to work out the magnification, you would do the size of the image that you're looking at divided by the size of the real object. So for example, if you wanted to know the magnification of this image, you'd need to use a ruler to measure the length of a cell, and then you would have to know the length of that actual cell as well. So in an exam question, they would have to tell you the actual length. You would then have to use your ruler to measure the image size. But it's key that you have them in the same units, because cells are often measured in micrometers, whereas you'll be measuring in millimeters. So here's a little conversion, which again you might find helpful to put onto a flashcard, because it's letting you know if you wanted to convert millimeters into micrometers, you would need to times by 1,000. And if you wanted to convert micrometers back into millimeters, you'd need to divide by 1,000. So make sure you have these in the same units, and then that would give you your magnification. Sometimes you might have to work out the size of the real object, and in which case you'd rearrange this formula, and that would then be size of image divided by magnification. You might also have to do scientific drawings. This is one of the skills. And this is when you are taking observation of structures from under the microscope and representing it as a scientific drawing. And there's a few key features that you need to know about scientific drawings. I do actually have an entire video on scientific drawings, which I'll link up here that you can watch. But this is in summary what you would need to make sure you are following in terms of the rules. It should be drawn in pencil. You should have a title for the diagram and indicate what the specimen is within that title. If the magnification is known, which hopefully it is, if you're doing it on a microscope, then you should state the magnification. You should always annotate the cell components, the cell sections, any tissues visible, just to show what you are visualizing. There shouldn't be any sketches, and what that means is you should only use solid lines, no overlapping lines. It's not an artistic drawing, it's a very factual drawing just showing shape, proportion, location. And there shouldn't be any colour or shading in either. So that's the whole aim of a scientific drawing, showing the size, location and proportion. Now you might have to use an eyepiece graticule as well to be able to measure the actual size of the object that you're viewing under the microscope. And to do this, you need to calibrate your eyepiece graticule using a stage micrometer. So inside the light microscope, there is a scale on a glass disc which is inside of the microscope, and that is called the eyepiece graticule. So it's within the eyepiece, which is the long part that you look down, and you use this to measure the size of objects that you're viewing under the microscope. But as I said, you need to calibrate it each time, because as you're looking at this scale in the eyepiece, you need to know what each one of these divisions is worth at each magnification. So you'd place a micrometer, a stage micrometer, within your view to calibrate it. So let's go through how you'd actually calibrate it then. So step one is you need to line up your stage micrometer, which is the ruler that you would put on the stage of the microscope. Line that up with the ruler, which is the eyepiece graticule, which is already within the microscope. So that means when you look through your eyepiece, you should now see two rulers and they are lined up. So we've got our eyepiece graticule lined up with the stage micrometer. And what you then need to do is count how many divisions on the eyepiece graticule fit into one division on the stage micrometer. And the divisions on the micrometer are known distances. So each division on the micrometer is 10 micrometers. So this can then be used to calculate what one division on the eyepiece graticule is worth at that current magnification. So for example here, one division on the micrometer is 10 micrometers. And we can see here that two divisions fit into one of those stage micrometers. So this top one is the eyepiece graticule, the bottom one is the stage micrometer. We know that one division is worth 10 micrometers. Two eyepiece graticule divisions fit into that. So we'd do 10 divided by 2, which gives 5 micrometers. So that means at this magnification, one division on the eyepiece graticule is worth 5 micrometers. And you would then take away the stage micrometer ruler, and then you'd put in your slide, and you can then measure cells using your eyepiece graticule when you now know what each division is worth and measure the actual size of the images that you are viewing. Then we go on to the electron microscopes in a bit more detail. So a beam of electrons is what is creating the image, and they have a very short wavelength, and that is why you have a high resolution. And because the resolution is so high, that means small organelles and internal structures can be viewed. The image is created by condensing that beam of electrons into a really fine beam, and that is using an electromagnet. That is what focuses the beam of those negatively charged electrons. Now, electrons are absorbed by air, and this is why when you're using an electron microscope, it has to be a vacuum, so there's no air present. You've just got your specimen there. And for that reason, you can't view living specimens, because if it's a vacuum, that's going to cause the specimen to die. So you can't use living specimens, which is a downside. And also you only get black and white images, and you have to stain the sample. Now, we did talk about some differences between the transmission and the electron microscope. We're going to go into it in a bit more detail. For a transmission electron microscope, because the electrons are transmitting or passing through the specimen, that means the specimen has to be sliced extremely thin, and you'd need to stain it as well, then you put it in a vacuum. The electron gun will then be fired at the specimen to produce a beam of electrons. That is then going to be focused using the electromagnet, and some parts of the specimen absorb the electrons, and that is why they appear darker on your image. So you end up with a 2D image like this one, where you can see the internal structures of an organelle. And this here is showing you the internal structure of a mitochondria. The scanning electron microscopes don't need to be as thin because the electrons aren't passing through. They are instead reflecting and scattering off the surface. And because they're scattering off the surface, that's how you get these different contours and therefore this 3D image of the surface of your specimen. So that's what you need to know about microscopes. The next part of cell structure is knowing this part 1.2, cells are the basic units of living organisms. So here is an extensive list of all of the organelles you need to know about in eukaryotic cells. And you need to know the structure and the function of these. And I've split it into the top part is what you'll find in animals and plants. The bottom ones that are highlighted in green, you would only find in plant cells. So let's go through the structure and function of each of these. One thing you could be asked to do as well is to identify the organelles within a cell. So here we have an image which is showing us an animal cell. The cell membrane is that outer layer. We do have a vacuole labelled here, but animal cells don't have permanent vacuoles. They can have temporary vacuoles. So for example, a phagocyte, which is the white blood cells that engulf pathogens, they trap the pathogen after it's been engulfed inside of a phagosome, which is a temporary vacuole. We then can see here our rough endoplasmic reticulum, so we can see those folded cisternae with the ribosomes on the outside. The Golgi apparatus does look very similar to the smooth endoplasmic reticulum, but the key difference is the Golgi apparatus curves, so it looks more like, often people tell me it looks like a Wi-Fi symbol, so you can see it curves a bit more, and often you'll see vesicles budding off the edges as well. That's how to tell apart the smooth endoplasmic reticulum and the Golgi apparatus. Ribosomes are very, very small, so you're looking for very small spheres. Lysosomes are larger than the ribosomes. Mitochondria, you have these inner foldings, which helps those to be identified as well. Then if we have a look at the plant cell, so all of the same organelles plus some extra. So we've got the chloroplasts, which do look similar to mitochondria because they've got these inner foldings as well. We've got a permanent vacuole, and the membrane on the outside of that is called the tonoplast. And then in the cell wall, that's an extra layer on the outside, and the pores in the cell wall are the plasmodesmata. But let's go through each of those structures, looking at the structure and function in more detail. Starting with the cell surface membrane. Cell surface membranes are found in all cells, and it's made up of this phospholipid bilayer, meaning you've got two layers of phospholipid, where you have the hydrophilic heads on the outside and the hydrophobic tails pointing inwards. You have molecules embedded within that as well, which are typically proteins and cholesterol. The proteins could be these protein channels, which enable molecules to pass through, or protein carriers, which also allow molecules to pass through. You also get glycoproteins, which is where you have a protein embedded within it and a carbohydrate attached, or you can get as well other molecules such as a glycolipid, which is where you have a carbohydrate attached to the phospholipid. And those roles for those two molecules are often things like receptors. So the function of the plasma membrane we'll learn more about in the topic, which is looking at membranes and controlling what can enter and exit. But that is what the role is. It can control what can pass through the membrane, meaning what can enter and exit a cell. Next, then we look at the structure and function of the nucleus. So the nucleus is a membrane-bound organelle, and that membrane is the nuclear envelope, which we can see here. There are pores, which are tiny holes within that nuclear envelope, and that's how mRNA, which is created inside of the nucleus, is able to leave the nucleus to go to the cytoplasm. Inside of the nucleus, there is nucleoplasm, which is a granular jelly-like material. It's also where you find the chromosomes, and in eukaryotic organisms, chromosomes are protein bound, so they're attached to histone proteins, and we describe the shape as linear. You have inside of the nucleus as well a nucleolus, which is the smaller sphere inside, and that is where RNA, specifically rRNA, is made, and rRNA is used to make ribosomes. So the function then, it is the site of DNA replication. It's also the site of transcription, which is the first stage in protein synthesis, which results in the creation of mRNA. It's also the site of rRNA production and ribosome synthesis, and it contains the genetic code for each cell. Flagella is the next structure, and this is a whip-like structure we can see here, and this will spin round at the base using a motor, and as it spins, it enables movement. Now, you don't have this on every single cell, but one example of a cell that would have it is a sperm cell, for example. Cilia, again, aren't on all cells, but some cells do have cilia, and these are hair-like projections coming out of the cell. Now, these cilia can be mobile or stationary. Mobile cilia help move substances in a sweeping motion. So the cells lining your trachea, which is part of your breathing system connecting to your lungs, have cilia, and they are mobile cilia to sweep and move mucus up and out of the trachea, so it's to prevent lung infection basically. Stationary cilia are important in sensory organs as well, so for example in the nose. Microvilli, again, aren't on every single cell, but some cells do have microvilli, and these are folding in the cell surface membrane to create these finger-like projections. And the function of this is it increases the surface area for transport across the membrane. So, for example, the epithelial cells lining the ileum have microvilli, and that increases the surface area to maximise the absorption of molecules like glucose and amino acids after digestion. Also, within the proximal convoluted tubule in the nephron of the kidney, there are microvilli to increase the surface area to maximise the reabsorption of glucose. Centrioles and microtubules are the next organelles we're going to have a look at, and centrioles are made up of microtubules, and they occur in these pairs to form what's known as a centrosome. The function is to produce, or they're involved in the production of spindle fibres, which are released to organise the chromosomes into particular positions during cell division. The microtubules in particular also aid with the movement of cilia and flagella. The cytoskeleton is the next one, and this is a network of fibres found within the cytoplasm all over a cell. And it consists of microfilaments, microtubules and intermediate fibres, which we can see here. The function is it provides mechanical strength to cells, it helps to maintain the shape and stability of a cell, and many organelles are bound to the cytoskeleton also. So we can see that here, that organelles are actually attached to this cytoskeleton. The microfilaments within it are responsible for cell movement. The microtubules are responsible for creating a scaffold-like structure. And then the intermediate fibres provide some mechanical strength. Next then we have a look at the endoplasmic reticulum, and you have rough and smooth endoplasmic reticulum. Both have folded membranes which are known as cisternae, and we can see those here. The difference then is the rough have ribosomes attached on the outside, and therefore the function of the rough endoplasmic reticulum is protein synthesis because it's got these ribosomes on the outside. And the smooth endoplasmic reticulum, their function is to synthesize and store lipids and carbohydrates. The Golgi apparatus, which is shown here in this peachy orange colour, again it's made up of folded membranes which are known as cisternae, but it's more of a curved shape. And you also get these secretory vesicles that pinch off. And that is because anything that's been processed and packaged within the Golgi apparatus is then secreted out in these vesicles. So within that Golgi apparatus, the functions are you'll have carbohydrates being added to proteins to form glycoproteins. They might produce secretory enzymes, they're going to secrete carbohydrates, transport, modify and store lipids, they might form lysosomes. Molecules are labelled for their destination here as well, and they get sent off in those secretory vesicles. And that's how the finished products are transported in these vesicles, which then fuse with the cell surface membrane, which then means they're released from the cell. Lysosomes are our next organelle. Lysosomes are the next organelle, and these are bags of digestive enzymes, and they can contain 50 different digestive enzymes. So they will hydrolyze phagocytic cells. That's one example of a lysosome that you learn about in phagocytes. They can completely break down dead cells, which is known as autolysis. You also get exocytosis, which is where they can release enzymes to the outside of the cell to destroy material. And they can digest worn out organelles for reuse of materials as well. Next, then we have a look at the mitochondria, and this is a double membrane bound organelle. The inner membrane is highly folded, and we call it the cristae. Inside of that, we have a fluid center called the mitochondrial matrix, and that contains ribosomes, the smaller ones, which are 70S, and it also contains loops of DNA so that it can create its own enzymes. So this is the site of aerobic respiration, therefore the site of lots of ATP being produced, and as I already said, it has DNA so it can code for its own enzymes required for respiration. Ribosomes are our next organelle, and these are made up of two subunits. We have a large and a small subunit. A ribosome is made up of protein and rRNA, and there are two types of ribosomes. ATS are the larger ribosomes found in eukaryotic organisms. 70S ribosomes are the smaller ones that prokaryotic cells have, but it's also the ribosomes that you find in the mitochondria and chloroplasts of eukaryotic organisms. And the function is it's the site of protein synthesis. We then move on to the organelles that are just found in plants. So chloroplasts, this is another double membrane bound organelle. And then inside we have these folded membranes. So this is your thylakoid membrane, which is highly folded to create these stacks that look a bit like stacked up coins. And those stacks are known as the grana. And they are embedded, those membranes are embedded with lots of pigments and proteins which are needed for the light-dependent reactions in photosynthesis. In peach colour here, that is a fluid centre, which is known as the stroma, which contains lots of enzymes that are needed for the light-independent reactions of photosynthesis. So that's the function of this organelle, it's the site of photosynthesis. The cell wall is found in plant cells, but also fungi. Fungi are also eukaryotic organisms. You do have cell walls in prokaryotic organisms as well, but we're just going to focus on cell walls of the eukaryotic organisms for now. So plants have a cell wall made up of cellulose, whereas fungi have a cell wall made up of chitin, which is a nitrogen-containing polysaccharide. And the function of the cell wall is to provide structural strength. Plant cells also have a large permanent vacuole, and this is a single membrane bound sac. For example, we've got the phagocytic vacuoles in animals, but those are temporary, and we're just focusing on the permanent vacuoles in plants. And those have a tonoplast, that's the name of the membrane that surrounds them. The plant vacuole's function is to regulate osmosis, and also it contains the pigments which give flowers their colour, so it's going to attract pollinators. The plasmodesmata is the next structure, and these are pore-like channels within the cell wall, which we can see here, and that provides a channel between cells to enable the movement of water and dissolved mineral ions between cells. So we then go on to photomicrographs, which is how you can identify these different organelles from images taken under the microscope. And a photomicrograph is an image that's been captured by a light microscope. They offer a lower resolution because of everything we said about the microscopes. So the light microscope, the image is created using light, which has a longer wavelength of light. But what you can work out from this is, in plant cells, you might be able to identify features such as the cell membrane, chloroplasts, and the vacuole. In animal cells, you'll be able to see key structures like the cell membrane, nucleus, and cytoplasm. But to be able to see other smaller organelles and the internal structure of organelles, that's when you'd need to use your electron micrograph or an electron microscope to create an electron micrograph. That's the name of the image that is created using an electron microscope. And because those microscopes have a higher resolution, that's why you're able to see all of these extra organelles and also the internal structures of those organelles. So if we move on to prokaryotic cells next then, and look at the key differences between the prokaryotic cells and eukaryotic cells. So first of all, prokaryotic cells are much smaller. So typically they're 1 to 5 micrometers in diameter. They don't contain any membrane-bound organelles. They do have ribosomes, but it's the 70S smaller ones. They contain DNA, but not within the nucleus. They do have a cell wall, but it's made of a molecule called murine. Some prokaryotic cells also contain plasmids, a slime capsule around the outside, and flagella, but they don't all contain those. So let's have a look then at those differences. They don't contain any membrane-bound organelles. So that means they don't have any mitochondria, chloroplasts, endoplasmic reticulum, Golgi apparatus, lysosomes, but they will have circular DNA loose within the cytosol, which is the name of the cytoplasm. They do have ribosomes, as we said, but it's the smaller 70S ribosomes. They don't have a nucleus. Instead, their DNA is circular and it is loose within that cytoplasm. We did say some of them contain plasmids, and plasmids are small loops of DNA which only carry a few genes. And you can get varying numbers of these in your prokaryotic cells, which are typically bacteria. The cell wall, we said they do have, but it's made of a glycoprotein called murine. And that slime capsule, the capsule on the outside of the cell wall that is only sometimes there, it's made of a protein, and the function is it prevents the bacteria from drying out or desiccating, and it also helps to protect the bacteria against the host's immune system because it will help to partially cover the antigens. The flagella, again, that's only sometimes present on bacteria. That can rotate and it helps to make the bacteria move. So this would be a good table to copy down to have in your revision to compare the structures that you'd find in a prokaryotic organism and eukaryotic organisms, but also split into the plants and animals. Lastly then is known about viruses, and viruses are non-living and non-cellular, so we often describe them as virus particles. So they are even smaller than bacteria, and they only contain a nucleic acid core, which could be DNA or RNA, a capsid, and make sure you do not confuse this with the capsule that's found on some prokaryotic organisms, because the capsid is this part here that surrounds the nucleic acid core, this protein layer. On the outside, there are attachment proteins, which are used to attach to receptors on the host cell. And then some of them have an outer envelope made up of lipids, which would be phospholipids. Viral replication occurs inside of host cells, and it involves the injection of that nucleic acid into the cell. And bacteriophage are an example of a virus that can infect bacteria. Next then, we move on to the topic biological molecules, which is split into these 4 components, and we're going to start with testing for biological molecules. So the first biological molecule that we're going to look at for testing is starch, and this one's quite straightforward. You would add the chemical iodine, which is an orangey brown colour, and if starch is present, it goes blue-black. The next then is to test for reducing sugars, and there's a bit more to it for this one. First of all, you need to add Benedict's reagent, which is this blue colour. But for this one, if you don't heat it up, the reaction won't happen. So the mark schemes will be really specific that you have to say add Benedict's reagent and heat. After heating, a positive test result will mean that that blue colour will change to one of these colours. And the more red it is, the higher the concentration of sugar present. So here we can see this orangey colour, so that would be a medium-high concentration of reducing sugar present. You can also use reagent test strips, and those can be used to test for the presence and concentration of reducing sugars. Then the test for a non-reducing sugar, so for example sucrose, you would still have to do your Benedict's test to start with, and if your sample remained blue, then you move on to the next stages, but there's still a mark for saying following a negative Benedict's test. You would then add acid and boil, and it does have to be boiling hot so that you can hydrolyse sucrose into fructose and glucose, so you'd have to say boil. You then need to cool the solution and add an alkali to neutralise, and then you would add Benedict's reagent and heat, and then you should get a colour change if a non-reducing sugar was present, and it will go, it says here blue to green, yellow, orange or brick red, but typically it actually goes orange or brick red, because if you're testing and you do have a reducing sugar, because you've hydrolyzed that disaccharide into monosaccharides, you've doubled the concentration of sugars present, so it should go brick red. Then we've got the semi-quantitative Benedict's test. So you'd prepare your Benedict's reagent, and you would then add this to a known concentration of glucose and note down the time it takes for that colour change to occur. So to prepare the sample, you could dilute the reducing sugar solution to a known volume with distilled water. So you can link this to the skill of serial dilutions to create a series of different concentrations, known concentrations of glucose. And for each one, you'd perform the Benedict's test, time how long it takes for each of those concentrations to change colour, and then you'd note that down, and if you plotted that as a graph, the time taken for each one to change colour, then it is semi-quantitative. The test for proteins then would be adding biuret, and the reason I've split it up into biuret is to try and help you remember the spelling, because often students get this confused with burette, which is a piece of apparatus you use in chemistry for titrations, and the spelling does matter, biuret, and that is this pale blue colour. You would add that to your sample, you do not need to heat, and if there is protein present, it turns this purple lilac colour. Then we have the test for lipids. The first step would be you need to dissolve your sample in ethanol, and to do that you would add ethanol and shake. Once you've done that, then you would add distilled water, and if you have a lipid present, you'd get this white emulsion. And you do have to state the colour, which is white, and emulsion is the description of the fact that you've got this thicker substance. It's not a precipitate, but it's what we call an emulsion. Next then, we move on to carbohydrates and lipids. So you need to know the difference between a monomer and a polymer. So monomers are smaller units which can bond together to create larger molecules, and a polymer is made from lots of monomers bonded together. And you need to know about the following monomers and polymers. So we're going to go through those, but you also need to know about macromolecules, and those are giant molecules. There are three that you need to know: polysaccharides, which are the top three listed here, proteins, and then the polynucleotides, which form in DNA and RNA. So if we have a look at the carbohydrates first of all, carbohydrates are made up of carbon, hydrogen and oxygen, and they can be categorised into monosaccharides, disaccharides and polysaccharides. Mono means one, saccharide means sugar. So a monosaccharide is the monomer, that's when you just have one sugar unit. A disaccharide is a dimer, which means two units joined together, and di means two, saccharide means sugar, so it's two sugars joined together. Polysaccharides are the polymers, poly meaning many, or in this case it literally means more than two, and that's when you have many sugar units bonded together. And here are some examples of each of those that you need to know. So if we have a look at glucose, alpha glucose to start with, this is the key monosaccharide that you will learn about, glucose. And the formula is C6H12O6, and here we have alpha glucose, the structure that you would need to know. Glucose has two isomers though, which means two forms of glucose that have the same molecular formula, but a different structure. And we can see here we've got alpha glucose on the left, and on the right we have beta glucose. And the key difference is alpha glucose, I always think it's pretty much symmetrical. You've got hydrogen on top and the hydroxyl on the bottom. That's the same on this side over here. Whereas for beta glucose, you have the hydroxyl on top and the hydrogen on the bottom. And that slight change in the position of the hydrogen and the hydroxyl has a big impact on the location and the type of bonds that can form when creating the polysaccharides, and therefore the final shape of the polysaccharides. So monosaccharides, these can be categorized according to how many carbon atoms they contain. A hexose sugar, hex means 6, and wherever you see OSE at the end, that means it is a sugar. So hexose means it's a sugar that contains 6 carbons. Pentose means it's a sugar that contains 5 carbons. For example, ribose. The disaccharides, these are made of 2 monosaccharides bonded together with a glycosidic bond. They're formed via a condensation reaction. And the key ones that you need to know are these here: maltose, lactose, and sucrose. All 3 of them contain glucose as one of the monosaccharides, but it's the second monosaccharide that differs. So maltose contains 2 molecules of glucose, lactose contains glucose and galactose, and the way I always remember that is there's lactose within the name, and then sucrose is made of glucose and fructose. So when you join those together, because it's a condensation reaction, you form a bond between those molecules, and that also involves the removal of water, which is why we've got plus water for all of those. So we said it's a condensation reaction that forms them. A condensation reaction is when you join 2 molecules together by removing water, and that forms a chemical bond. Hydrolysis is the opposite reaction. It's when you split apart molecules through the addition of water, which breaks a chemical bond. And we see these 2 reactions creating all of the polymers and also hydrolyzing the polymers back into their monomers. So here it is in action. We're just showing glucose, but without any of the hydrogen or hydroxyl groups, except for the ones involved in the condensation reaction. So here we have 2 molecules of glucose, and the water is going to be removed from these hydroxyl groups. It doesn't actually matter which way round the full hydroxyl and then the single hydrogen is removed from. The point is that you're left with a single oxygen atom behind. And that is what the glycosidic bond is, the carbon to oxygen to carbon. So that's the bond that is formed to join these 2 molecules together, and it involved the removal of water. And we'd call that a 1 to 4 glycosidic bond because the bond is formed between carbon 1 and carbon 4. And we always number the carbons going clockwise from the oxygen in the ring. So carbon 1, 2, 3, 4, 5, 6. And again, carbon 1, 2, 3, 4, 5, 6. So the bond is formed between carbon 1 on this glucose molecule and carbon 4 on this glucose molecule. A hydrolysis reaction then would involve adding that water back in to break that glycosidic bond, and then we'd get those 2 hydroxyl groups back, and we now have our monosaccharides again. So the polysaccharides, these are created by repeated condensation reactions between many glucose monomers. Starch is found in plants, and its function is a store of glucose. Cellulose is also found in plants, and that is a structural molecule, so it provides structural strength to the cell wall. And glycogen is found in animals, it's also a store of glucose. So glycogen and starch are very similar in structure because they have the same function, it's just one is found in plants, one is found in animals. So here is the summary table. This would be a good one to screenshot and print out, or to write it out, so you've got your summary of the structure and function of all 3 of those polysaccharides. So the first one we've got is which isomer of glucose the monomer is in all 3. So starch and glycogen both have alpha glucose, whereas cellulose has beta glucose. And it's that key difference which explains why cellulose can only form 1 to 4 glycosidic bonds, whereas alpha glucose enables the formation of 1 to 6 glycosidic bonds as well. And when you have a 1 to 6 glycosidic bond, that causes a branch to come off the structure. And that is why starch and glycogen, if we jump to the structure part, they both are branched molecules. Now that is an advantage for starch and glycogen because the fact that they are branched means that there is a greater surface area to these molecules. So therefore, when enzymes attach to hydrolyze that store of glucose back into glucose, they can more rapidly hydrolyze that molecule. And that's good because it means you get the glucose being released for respiration more rapidly. All three molecules are very large, so they're insoluble, which means they won't dissolve, won't affect the water potential or osmosis in the cell. Cellulose is a very different structure. This is the one that we said is made of beta glucose. 1 to 4 glycosidic bonds joins them together, and that means you don't have any branches. You just get these long, straight chains of the polymer, which can therefore lie really closely together in parallel, and hydrogen bonds then form between these chains. And the fact that so many hydrogen bonds form between them is what provides this structural strength to the molecule. A single hydrogen bond is actually very weak, but because there are so many holding together your chains of beta glucose, which is then called a fibril or a microfibril or a macrofibril, that is what provides the structural strength. The lipids was the other part of this submodule, and these are macromolecules, but they are not polymers. You do not have repeated units bonded together, so it is still large, but it's not a polymer. They're non-polar molecules, and for that reason they're insoluble in water, and they will only dissolve in organic solvents such as ethanol. They are hydrophobic, which means they will repel or they won't interact with water. And they're made up of 3 fatty acids for a triglyceride and a glycerol molecule, and for a phospholipid it's 2 fatty acids and a glycerol plus that phosphate group. But what they all have in common is they're made up of glycerol and fatty acids. So these are the 2 key lipids that you will need to know the structure of, and the main difference is that a phospholipid has only 2 fatty acids, so instead of that 3rd fatty acid, it's got a phosphate group attached to the glycerol, and that completely changes the properties of the lipid. So how a triglyceride is made is actually the same way that a phospholipid is made. Your glycerol and fatty acids join together through condensation reactions, which we said was the joining together of molecules, forming a chemical bond, removing water. And it's at this point here between the glycerol and the fatty acids that the water molecule would be removed. And if it's a triglyceride, you have 3 condensation reactions, so 3 molecules of water are removed, and we then get 3 bonds, and those bonds are known as ester bonds. And that is an R, your R group here is the fatty acid hydrocarbon chain, and then we've got the C double bond O, O, and the H is over here. So that would be your ester bond. This is just showing you in more detail how that condensation reaction happens between the glycerol and those fatty acids. So you'd have a molecule of water being removed, and that then leaves you with the triglyceride and your ester bond. And 3 molecules of water would be removed. Now the fatty acids can either be saturated or unsaturated, and what we mean by that is whether they contain a single or double bond between the carbon atoms in the R group, which is that hydrocarbon chain. So this top one, we can see only single bonds between the carbon atoms, so that would mean it's saturated because it's holding the maximum number of hydrogens possible, that's why it's saturated. An unsaturated fatty acid, you have at least 1 double bond between the carbon atoms. So it's not fully saturated, it could have held 2 more hydrogens here. And where you do get that double bond between the carbons, it causes a kink in the chain. We can see here it's bending. So the properties then of triglycerides, they can transfer energy, and that's because of the large ratio of energy storing carbon to hydrogen bonds compared to the number of carbon atoms. A lot of energy can be transferred when it's broken down. Also, due to the high ratio of hydrogen to oxygen atoms, they can act as a metabolic water source. And this is because triglycerides can release water if they are oxidized. And that's essential for animals such as camels in the desert. Also, because the lipids are large, they're hydrophobic molecules, which means they're insoluble, so they're not going to affect the water potential or osmosis. They also have a relatively low mass, and that means compared to other molecules of their size, such as a protein, they do not have anywhere near as high a mass. So you can actually store a lot of them in an animal without increasing the mass as much, which would prevent movement. Phospholipids, on the other hand, so we've talked about the fact that those are made of a glycerol molecule, 2 fatty acid chains, and a phosphate group. That means that they would only require 2 condensation reactions to join those fatty acids on, and you get 2 ester bonds. The properties are different though because of the phosphate group on what we describe as the head of the phospholipid. So the head is the glycerol and the phosphate group, and the phosphate group has got a negative charge to it. And because it's got a negative charge, that changes the properties and it makes it hydrophilic, which means it can interact with water. The tail is the hydrocarbon chains, or those fatty acid chains, and those don't have a charge, so they are hydrophobic, which means they will repel water, but they can interact and mix with lipids or fats. And for that reason, you get this phospholipid bilayer structure forming with lots of phospholipids. And that's because those hydrophilic heads can interact with water, but the tails are repelled by water. So if you were then to put large amounts of phospholipid in water, you will get the heads on the outside interacting with the water and the tails spin inwards, repelling away. So this behaviour of the tails moving away from water is what results in that phospholipid bilayer, which forms the plasma membrane around cells and some organelles. And the hydrophilic nature of the phosphate head enables that surface of the plasma membrane to stay in place. And the phospholipid bilayer arrangement enables carbohydrates also to attach and form important receptors on the membrane, and that would be your glycolipids. So next then we're going to move on to the proteins. So proteins are made up of one or more large polymers, creating a macromolecule. And the monomer that they're made up of are amino acids. So here is your general structure of an amino acid. You have NH2, which is the amine or amino group. You have your carboxyl group, which is COOH. You have a central carbon, and then we have an R group, which is the bit different on all 20 amino acids. So R means it's variable. And then you have a hydrogen. So the way that I always remember how to draw it is, remember you have this central carbon in the middle with 4 different groups coming off it. Then you have to try and remember the groups, your amine group, carboxyl group, R and hydrogen. So proteins are polymers. They're these macromolecules made up of the monomer amino acids. And there are 4 levels of structural organisation. And we're going to describe each of these, starting with the primary structure. So straight after protein synthesis, you have your primary structure, which is just a sequence of amino acids in a polypeptide chain. And you have to say either the sequence or the order, because that exact order of the amino acids is going to determine the bonding location that happens in the later stages. So it is the order or the sequence of amino acids held together by peptide bonds. That primary structure then folds or coils to make either a beta-pleated sheet or an alpha helix. And those are held in place by hydrogen bonds. So describing the structure of the secondary structure of a protein would be a 2-mark question. The first mark would be for saying that you get these alpha helixes or beta-pleated sheets. Second mark is saying they're held in place by hydrogen bonds. The location of those hydrogen bonds then is between different amino acids, and it's always between a hydrogen atom and an oxygen atom. Next then we have the tertiary structure, and this is the further folding, and it's held in place by a range of different bonds. You have hydrophobic and hydrophilic interactions, which are quite weak. You have even more hydrogen bonds, which are also weak. But then you'll have ionic bonds, which are slightly stronger. And the ionic and disulfide bonds, the disulfide bonds being the strongest of them, the covalent bonds, those are going to form between the R groups of different amino acids within the polypeptide chain. Now, you actually only sometimes get disulfide bonds, because disulfide means it's a bond between two sulfur atoms. And that means you're only going to get this bond if you have the R group, which contains sulfur, because sulfur is not in the rest of the general structure. So that tertiary structure then, it's the further folding to create a unique 3D shape held in place by those bonds. So it'd be a 3-mark definition. The further folding is 1 mark, unique 3D shape is a second, and naming the bonds is the third. And the location that those bonds form is determined by the sequence of amino acids in the primary structure. And where those bonds form is what determines the final unique 3D shape. So that's why the primary structure is so important. That sequence of amino acids determines the location of these tertiary structure bonds, and that determines how it folds and the 3D shape it makes. The last level of organisation is a quaternary structure. So some proteins, not all, are made up of more than one polypeptide chain. So you still get that 3D unique shape of the tertiary structure. It's just you have multiple polypeptide chains bonded together. So for example, hemoglobin is made up of 4 polypeptide chains. And hemoglobin has 4 polypeptide chains. Each chain has a prosthetic group attached, which means a non-protein group attached to the polypeptide chain. And that prosthetic group contains iron. So the group is called a heme group and it contains iron. So we then have a look at fibrous versus globular proteins. So we've talked about the fact that the tertiary structure is this 3D unique shape because of that further folding. But the way it folds, you can either get spherical shapes, which would result in globular proteins, or more rope-like shapes, which would be your fibrous proteins. So the fibrous proteins, that is when it folds to create these long twisted strands. And because of that, it creates a really stable structure, which is insoluble in water. And the function would be to give structural strength. So collagen, for example, in bones, or keratin in your hair. In contrast, the globular proteins, they fold up into these sphere-like shapes, which means they're relatively unstable. They are soluble, and they are involved in physiological functions. So, for example, all the enzymes, antibodies, and some hormones in hemoglobin. But because they're relatively unstable, these are all very temperature and pH sensitive. So let's have a look at hemoglobin in a bit more detail. We said it's a globular protein. It also has a quaternary structure. It's got 4 polypeptide chains, 2 that are called alpha chains and 2 that are called beta chains. It has a prosthetic group attached to it, and that is the heme group, which we can see here. It's described as a conjugated protein. Conjugated means it has a prosthetic group added to it. And that prosthetic group is the heme group, which contains iron, and it's the iron that the oxygen is going to be able to bind to. Collagen was our example of a fibrous protein. So collagen molecules are triple helices composed of 3 polypeptide chains wound around each other, like we can see in this diagram here. And these chains are held together by hydrogen bonds and some covalent bonds. The fibril formation then is that the collagen molecules interact with the adjacent molecules next to them, and that's going to form these crosslinks between their R groups, and they can then align in parallel. We get these staggered ends which prevent weak spots in the fibril. So overall it creates a tensile strength. So collagen's unique structure provides both flexibility but also tensile strength. So tissues like your Achilles tendon in your ankle is rich in collagen fibres, so it can withstand significant pulling force without stretching or breaking. And the collagen fibres can align differently based on the forces they encounter. In tendons, they form parallel bundles along the direction of the tension. In skin, layers of collagen fibres run in various directions to resist the pulling force from multiple angles. So the final biological molecule is water. So water is a polar molecule due to the uneven distribution of charge. So you have two hydrogen atoms and oxygen. The oxygen has a slight negative charge. The hydrogen atoms have a slight positive charge. So that's what this delta negative and delta positive symbol are representing. And that is going to enable hydrogen bonds to form between different water molecules. And that's what we're shown here. We've got a hydrogen bond forming between the oxygen and the hydrogen of different water molecules. And it's the formation of those hydrogen bonds that provide the properties that we're going to be talking about of water. So the key properties that you need to know is that it's an important solvent in reactions. It has a high specific heat capacity and a large latent heat of vaporization. So if we start with the fact that it's a solvent, this is due to the polar nature of the molecule. Because the hydrogen atoms have a slight positive charge and the oxygen have a slight negative charge, that means that it can attract positive and negative ions when they're placed within the water. And that is what causes the dissolving, because the water can surround those ions. So this could be sodium chloride, for example, in this example, and the sodium would be the positive, the chloride would be the negative. So the oxygen, which is a slight negative, would be attractive to the positive sodium. That can then separate that lattice and surround the sodium ions, and the hydrogen can do the same thing to the chloride ion. And as it separates all of those, that is what dissolving means. So it's possible because of that polar nature, but it can't dissolve non-polar molecules, and instead non-polar molecules would actually repel the water. The cytosol in eukaryotic and prokaryotic cells is mainly water though, so this is really beneficial because it means many solutes can dissolve within the cytoplasm of the cell, and therefore reactions are going to happen more rapidly, but also it means molecules can be easily transported, dissolved in the solution. So that links to this idea of transport mediums as well. Because water is a good solvent, plants, for example, they will have mineral ions dissolved in the water, which is then transported up through the xylem. And not only that, because of the hydrogen bonds that form between water molecules, the water molecules stick together, creating cohesion. And that means water can move up the xylem in the transpiration stream as a continuous column of water. And that makes it much easier to draw the water up the xylem with all of those dissolved mineral ions within it, so that the cells further up can gain access to both water and mineral ions. It's actually the same idea in blood, not that there is this cohesion, but blood is made up of plasma, which is mainly water, and that means you can dissolve ions in it and glucose, and that can be easily transported around the blood in animals. Next then is the high specific heat capacity, and water has a high specific heat capacity because of the large number of hydrogen bonds that form between water molecules. And therefore it's going to require energy to break those hydrogen bonds to split apart the water molecules. And for that reason, a lot of energy is required to raise the temperature of water. So water acts as a temperature buffer. And this is an advantage, as internal temperatures of plants and animals should therefore remain relatively constant, even if the external temperature is fluctuating. And that should prevent enzymes from denaturing. It's also particularly important for any organism that is aquatic, meaning it lives within bodies of water, because that should mean that the temperature of the body of water that they live in should remain relatively constant as well. Then we move on to the large latent heat of vaporization. This means that a lot of energy is required to convert water from its liquid state to its gaseous state. So in other words, for evaporation. And again, that's because of the energy required to break the hydrogen bonds between the water molecules. And because a lot of energy is required to evaporate water, that means that it can provide a significant cooling effect. So when you sweat, water is being released onto the surface of your skin. The heat energy that's radiating from your skin is used to evaporate the water, and in doing that, large amounts of heat energy are being removed from your skin, and it provides a cooling effect. Next then we move on to module 3, which is enzymes, split into these 3 topics, and we're going to be starting with mode of action of enzymes. So enzymes are an example of globular proteins, and they are biological catalysts. The active site is specific and unique in shape due to the specific folding in that tertiary structure, which is determined by the primary structure and the location of the bonds in the tertiary structure. Due to this specific shape, they get an active site that is unique, which will be complementary in shape to only one substrate. So that means enzymes are specific, they'll only catalyse one particular reaction. And that could be intracellular or extracellular reactions. So for example, catalase is an intracellular enzyme inside of liver cells that breaks down hydrogen peroxide into oxygen and water, whereas trypsin is an extracellular enzyme in the small intestines that hydrolyzes proteins. So if we think about how they are speeding up these chemical reactions, it's because all reactions require a certain amount of energy before they occur, and that is known as the activation energy, which you'll be familiar with from chemistry at GCSE. When enzymes attach to the substrate, they lower the activation energy needed for the reaction to occur, and therefore they speed up the reaction. So there's 2 models that hypothesize how they lower the activation energy. The lock and key model is the one that suggests that the enzyme is like a lock with a fixed shape, and that the substrate is like a key, which is perfectly complementary in shape to that lock. So they fit together, and you create an enzyme substrate complex, and that will then result in the distortion of the substrate, lowering the activation energy. Now, they'll only bind together because of random collisions. The difference between this and the induced fit model is, in this instance, it's more like the analogy that the enzyme is like a glove and the substrate is like your hand. So that means that they're not perfectly complementary to start with, but as soon as they bind together, like your hand going into the glove, they then become perfectly complementary. So what this means is the substrate is almost complementary in shape to the active site. When it collides, it induces the enzyme to mold around that substrate. And in doing that, it puts tension and strain on the bonds in the substrate, and therefore less energy is needed to break those bonds. And that is how it lowers the activation energy. Now, this is the current accepted model. Because of research that's been done, they've realised that enzymes, proteins, are slightly flexible. So this would make sense that that is the case. And also it better explains how the activation is lowered, the activation energy is lowered. Now, you need to know a little bit about certain reactions. So investigating enzyme catalyzed reactions, starting with, let's have a look at the catalase reaction monitoring. So catalase, we already said, that's the enzyme that catalyzes the breakdown of hydrogen peroxide in the liver into water and oxygen. So you could be asked to investigate the progress of this reaction, where you can measure the rate of formation of oxygen over time. So you could do this by collecting the oxygen produced in a measuring cylinder or maybe a gas syringe, and at set intervals record the volume of oxygen being produced. Alternatively, you could use a data logger. That's going to be a more accurate way to measure the oxygen being produced. But what you would then do is plot the volume or the pressure of oxygen produced against time, and the graph can be generated to then visualize the rate of the reaction, because rate is the amount of product produced over a set period of time. The amylase reaction monitoring is the alternative option. So amylase is an enzyme that catalyzes the hydrolysis of starch, that's the substrate, into simpler sugars such as maltose, and then eventually maltase would hydrolyze that into glucose. To monitor the progress of this reaction, we can measure the rate of the disappearance of starch over time, and we'd need to use iodine for this. So you'd need to take samples of the reaction mixture at regular intervals and test for the presence of starch using iodine. As amylase would be hydrolyzing starch into maltose, once the starch is hydrolyzed, the iodine would no longer change blue-black and instead it'd remain orangey-brown. So you could time how long it takes for the reaction to occur by seeing how long it takes for the starch to be broken down, and therefore you don't get a blue-black colour anymore. And you could always use a colorimeter, as a way to quantify this. So a colorimeter then is an instrument used to measure the absorbance or the transmittance of light by a solution, but it's normally the absorbance. It works on the principle that certain substances absorb light at specific wavelengths, and that results in a change in the intensity of the light that can be transmitted through the solution. Enzyme catalyzed reactions often involve colour changes due to the formation of reaction products or the loss of a substrate. So you could measure this within a colorimeter. So you'd place your sample in a cuvette, which is the name of the plastic holder, put it into your colorimeter. You then press the test button, and that will cause a beam of light to shine through your solution. And then you can see how much was absorbed by the solution versus gets transmitted and picked up by the detector. And that will then give you a numerical value. So you get a quantitative result. Next, then we have a look at the factors that affect enzyme action. And the key factors that you need to know about are temperature, pH, enzyme concentration, and substrate concentration. Now, the first two link back to what we were saying in the proteins part of this lesson, whereby enzymes are globular proteins, and globular proteins are less stable and therefore they're sensitive to certain conditions, meaning that they are more at risk of having tertiary structure bonds being broken, and therefore they lose their unique 3D shape. And that's what temperature and pH cause to happen. If there isn't a high enough temperature, enzymes don't denature, but at lower temperatures, there's insufficient kinetic energy for the successful collision between the enzyme and the substrate. And therefore, you get fewer enzyme-substrate complexes, and the rate of reaction is lower. If you go above the optimum temperature, though, there will now be so much kinetic energy that it causes the bonds, such as the hydrogen bonds, to break. And that results in the protein unraveling and losing its unique 3D shape. That means that active site changes shape, and therefore enzyme substrate complexes can't form. And we'd say that the enzyme has denatured. It's a similar idea with pH, because if you have too high or too low a pH, it will interfere with the charges in the amino acids in the active site. And if you're changing the charges, that can cause the ionic and hydrogen bonds to break. That changes the tertiary structure and therefore changes the active site, and therefore the enzyme has denatured. So you won't get those enzyme substrate complexes, and therefore the rate of reaction is going to be lowered. Now, each enzyme will have a different optimal pH based on the location that they are functioning in. So most of them will be around neutral, but some are slightly more alkaline, and some, for example, different protease enzymes that work in the stomach, have an optimum significantly lower, maybe around pH 1 or 2, so very acidic conditions. Next, then we have a look at the substrate and enzyme concentration. This is nothing to do with denaturing now. It's all to do with successful collisions, whether there is an active site available or not. So if there is a low concentration of substrate, the reaction will be lower as there will be fewer collisions between the enzyme and substrates because they're less available. If you increase the substrate concentration, you'll therefore get an increase in the rate of reaction, because at that point here on the graph, substrate is the limiting factor. But at high substrate concentrations, the rate of reaction plateaus because all of the enzyme active sites will already be in use. So even if you add more substrate, there are no empty active sites for them to collide with. So the reaction is already going at its maximum rate. With the enzyme concentration, similar idea. At low enzyme concentrations, there's going to be a lower rate of reaction because there'll be fewer collisions between the enzyme and the substrate, so fewer enzyme-substrate complexes. So as you increase the enzyme concentration, there'll be an increase in the rate of reaction. But at high enzyme concentrations, unless you also have unlimited substrate being added, the rate of reaction is going to plateau as there'll be insufficient substrates to bind with all of those enzymes. So you'll end up with some empty enzyme active sites. The next concept is inhibitors, which can prevent enzymes from working. A competitive inhibitor is one example, And these are the same or very similar in shape to the substrate, and therefore they're complementary to the active site, and they can actually bind to the active site, blocking it, and therefore preventing the substrate from binding. And if you have an enzyme inhibitor complex, that prevents an enzyme substrate complex, and therefore it lowers the rate of reaction. Now, most competitive inhibitors are reversible, and what that means is if a high enough substrate concentration is present, then the substrate can actually knock the inhibitors out, and therefore you can get the enzyme substrate complexes forming and the rate of reaction will increase again. So reversible means the inhibitor can be removed. Non-reversible means the inhibitor cannot be removed. Non-competitive inhibitors are inhibitors that do not bind to the active site. They bind to a different binding site on the enzyme, which is known as the allosteric site. Now, this doesn't block the active site, but instead, when it does attach to the protein, it binds, it causes the protein to change shape, and therefore the active site changes shape, and the substrate will no longer be able to bind. So no matter how much extra substrate you add, you will not get any more enzyme substrate complexes because the active site changed. And that's why you get these shaped graphs with inhibitors. The solid line is showing you, without any inhibitor, this is the curve that we just talked about, showing the effect of adding the substrate. Eventually it plateaus because all of the enzyme active sites are in use. Competitive inhibitors, the curve is shifted to the right, indicating a lower rate of reaction. But at high enough substrate concentrations that would knock out the inhibitor, so you do end up at the same Vmax, which is the maximum rate of reaction for that enzyme reaction. Non-competitive inhibitors, though, there's a lower rate of reaction and it plateaus at a lower rate as well. And that's because it doesn't matter how much extra substrate you add, the rate won't increase because the active site has changed shape. Now, enzyme inhibitors, often what people think is, what is the point of an enzyme inhibitor? Now, sometimes they are just harmful, they are poisons, things like cyanide, for example. But some of them are actually very beneficial and naturally occur in our bodies. And it's this concept of end product inhibition. And what this means is the products of some reactions are actually reversible competitive inhibitors. And in that way, it controls when reactions are occurring to make sure cells don't overheat and that you're not wasting resources. Because if there's a lot of product present, that product acts as an inhibitor. So it will inhibit the enzyme at the start of a reaction, and therefore it means the reaction isn't going to occur while you have lots of product present, because you don't need that reaction to occur. But when that product gets used up, that means there'll be no more inhibitor present and the reaction will start again. So it's a way to control when reactions do and do not occur. So we saw on that graph this idea of Vmax, and Vmax refers to the maximum rate at which an enzyme catalyzes a reaction when the active sites of all enzyme molecules are fully saturated with the substrate. And at Vmax, the enzyme is functioning at its maximum capacity, and further increases in substrate concentration won't increase the rate of reaction. So Vmax is a key measure used to characterize enzyme kinetics and is determined experimentally by plotting reaction rate against substrate concentration until you see that plateau being reached. You then got Michaelis-Menten constant, or Km, and Km is a measure of the affinity of an enzyme for its substrate. It represents the substrate concentration at which the reaction rate is equal to half of the Vmax. So Km can be thought of as the substrate concentration required to achieve half maximum velocity. Enzymes with lower Km values have a higher affinity for their substrates as they reach half maximal velocity at lower substrate concentrations. So Km is derived from the Michaelis-Menten equation, which describes the relationship between reaction rate and substrate concentration. Lastly, then we have a look at this comparison of enzyme affinity. Km values can be used to compare the affinity of different enzymes for their substrates. Enzymes with lower Km values exhibit greater substrate affinity and are more efficient at converting substrate into product, even at low substrate concentrations. And conversely, enzymes with higher Km values have lower substrate affinity and require higher substrate concentrations to achieve maximal velocity. You also need to be aware of immobilized enzymes compared to enzymes free in solution. And enzymes can be immobilized, meaning fixed, within a matrix such as alginate, or they could be just free to move around within a solution. When investigating the difference in activity between immobilized and alginate or free in solution, experiments can be conducted comparing reaction rates under controlled conditions. So, for example, the activity of an immobilized enzyme can be compared to that of the same enzyme in a solution by measuring the rate of substrate being used up or product being produced over time. Now, the advantages of using immobilized enzymes are it makes them more stable. Immobilized enzymes are less sensitive to temperature and pH changes, and that makes them very useful to use in industrial reactions because you can use a slightly higher temperature to speed up the reaction without denaturing the enzyme. It also makes it much easier to separate the product from the enzyme, because here we have the enzyme immobilized in alginate beads. You could easily just sieve those out and then you have the product remaining in the liquid. You can have the continuous use of enzymes as well. Immobilized enzymes can be used continuously in bioreactors and industry as they're packed into columns or membranes and you just have a flow of your substrate going over them continuously. That means they're also easy to reuse, making it much quicker and also cheaper. And we've got here improved reactor performance. Immobilized enzymes can improve enzyme activity as well by providing a higher local concentration of substrate near the active site. So it increases the rate of reaction also. Next then we're going to have a look at cell membranes and transport, starting by looking at the fluid mosaic model of membranes. So all cells and organelle membranes are composed of a phospholipid bilayer. And this provides a partially permeable membrane so that you can control what can enter and exit the cell. It can also be the site of chemical reactions and have a role in cell communication. So the fluid mosaic model then is this concept that you've got the mixture and movement of the phospholipids, proteins embedded within it, glycoproteins, glycolipids, and also that cholesterol. So it's a mosaic because it's made up of different molecules, and it's fluid because there is some movement in this molecule, so left to right movement. The phospholipids align as a bilayer, and we talked about earlier in this video that that's due to the hydrophilic heads being attracted and interacting with water on the outside, and the hydrophobic tails are repelled so they spin inwards. Proteins within the cell membrane can be intrinsic, meaning they're called intrinsic or integral, and that means they go all the way through the membrane. Or they can be called extrinsic or peripheral, which means they're just on the outside of the membrane. The extrinsic proteins provide mechanical support, or they can make glycoproteins. They wouldn't be a glycolipid because that's not got a protein in it. The glycolipid would actually be these green parts which are separate, but the function of all of those is cell recognition as receptors as well. Intrinsic or integral proteins are protein carriers and channel proteins involved in the transport of molecules across membranes. Protein channels form these tubes that fill with water to enable water-soluble ions to dissolve and then diffuse through the channel, whereas the carrier proteins will bind with the ions and larger molecules such as glucose and amino acids and change shape to transport them to the other side of the membrane. Cholesterol is shown here in yellow, and that is present in some membranes, and this restricts that lateral, meaning the side-to-side movement, of other molecules in the membrane, and this is useful as it makes the membrane less fluid at high temperatures, and therefore prevents water and dissolved ions from leaking out of the cell. So cell signalling refers to the transmission of messages or signals from one part of an organism to another, and it ensures coordinated responses to internal and external stimuli. This process is vital for the proper functioning and survival of organisms, and the cell membrane plays a role in this. So the purpose of the cell signalling, all the cells and organisms must respond to their environments to be able to survive changes in the environment. And signalling pathways coordinate cellular activities. Those pathways could be electrical, for example, in the nervous system, or chemical, for example, in the hormone system, and they involve various molecules such as neurotransmitters and hormones. So the stages in the chemical signalling pathway, and this is where we'll see it link back to the role of the membrane, you have secretion of a specific chemical or a ligand. And cells secrete specific chemical messengers, and that's what the ligands are, in response to a stimuli. And that could be molecules like hormones, for example, glucagon, which is then going to be released into an extracellular space. That will then be transported through the bloodstream, in the case of hormones, to the target cell. And once it gets to the cell, the ligand will bind to receptors on the cell surface membrane of those target cells. And this is where we see the role of the cell membrane in the chemical signalling pathways, because they have those receptors on the cell surface membrane. That's what the hormone or ligand can bind to, and that will initiate a signalling cascade. So the cell surface receptors are protein molecules located on the cell surface membrane. The ligand receptor binding triggers the conformational changes in that protein receptor, allowing the message to be transmitted into the cell. And that process is known as transduction. It often involves G proteins and the production of a second messenger, so another chemical that's going to amplify the effect. Second messengers will relay the message by activating enzymes. And once an enzyme is activated, it's going to cause a chemical reaction to happen inside of the cell. So that sequence of events triggered by the binding to the receptor is known as a signaling cascade, as we just went through. And the receptors can alter cell activity by opening ion channels or acting as membrane-bound enzymes or serving as intracellular enzymes as well. And hydrophobic signaling molecules like steroid hormones can diffuse directly across the cell membrane and bind to receptors in the cytoplasm instead, or even in the nucleus. You also have other mechanisms of signalling. So you could have direct cell-to-cell contact using receptors on the cell surface membrane. So that's what we can see here, for example. Antigens embedded on the cell surface membrane. You're going to have a cell-to-cell contact through the antibody or an antigen-presenting cell and maybe a T helper cell. Next, then we move on to the movement into and out of cells, which is controlled by the cell surface membrane. And there are 6 key modes of transport that you need to know: simple diffusion, facilitated diffusion, osmosis, active transport, and then endo and exocytosis. So simple diffusion is the net movement of molecules from an area of higher concentration to an area of lower concentration until equilibrium is reached. This process does not require ATP. It doesn't require a membrane either, even though we can see that here. It doesn't always happen across a membrane. But we can see we've got a concentration gradient, and as long as the molecule is small and lipid soluble, it can then dissolve and diffuse across that membrane until equilibrium is reached. Facilitated diffusion is also a passive process, meaning it doesn't require energy from ATP, and that's because it's still going down the concentration gradient. But this would be used for molecules that are either not lipid soluble or they are large, and therefore they have to go through a protein instead because they can't dissolve through the phospholipid bilayer. Now this could be either using a protein channel or a protein carrier. So protein channels, for example, you would have a channel which is a tube filled with water, which would mean ions or polar molecules can dissolve in that water instead of the phospholipid bilayer and then move through that channel by diffusion. Carrier proteins is when you'd have the molecule complementary in shape to that particular protein bind. When it binds, it causes a conformational change, which results in the molecule being released on the other side of the membrane. Water moves across the membrane by osmosis. So your definition of osmosis is the movement of water from an area of higher water potential to an area of lower water potential, or more negative, across a partially permeable membrane. So there's different types of solution when we think about water potential. Isotonic is when the water potential, which is a measure of how concentrated a solution is the same outside of the cell as it is inside of the cell. So iso means equal. Hypotonic is when the water potential of the solution surrounding a cell is more positive, which means it's closer to zero, which in other words means it's less concentrated. So you don't have as much solute dissolved in the solution compared to the cell. Hypertonic is when the water potential of the solution surrounding a cell is more negative than the cell. So a hypertonic solution means that you have more solute dissolved in the water. And we can see here when you place animal cells, which don't have a cell wall, into these three different types of solution, the effect it has on the cell. So if you were to put red blood cells, our example of an animal cell, into a hypertonic solution, because the solution has a more negative water potential, the water is going to move from the cell to the surrounding solution by osmosis, and that causes the cell to shrivel up or crenate. Plasmolyze is a term we use if it's happening in a plant cell. If it's an isotonic solution, because you've got an equal water potential inside and out, there's not going to be any net movement of water. So some water will move in, some will move out, but the cell will stay the same size and shape. Hypotonic solution is when the inside of the cell is more negative compared to the solution in terms of water potential. So water is going to move into the cell by osmosis, which can cause the cell to swell, and if enough water moves in, it causes the cell to burst because there's no cell wall to provide that structural strength. Active transport is the movement of molecules and ions against the concentration gradient. So going from an area of lower concentration to higher concentration. And because it's going against the concentration gradient, energy is required from ATP, and also you need a carrier protein, and it is always carrier proteins, not channel proteins. Now this is a selective process because it's still linked to the idea of a particular molecule has to be complementary in shape to a specific carrier protein. So ATP is involved and it will bind to the protein on the inside of the membrane, and as it is hydrolyzed into ADP and PI, it's going to release energy. And that energy causes a change in shape to the protein to cause it to open out to the other side of the membrane. And as it opens up to the other side of the membrane, it releases the molecule to the other side of that membrane. The PI, which is the inorganic phosphate, is then released from the protein, and that causes that carrier protein to revert to its original shape, and as long as there's still ATP, that process can continue. Next time we have a look at endocytosis, and this is a type of active transport, and it is the bulk transport of molecules into a cell. So endo means in. The cell surface membrane will bend inwards around the molecules surrounding it to form a vesicle. The vesicle will then pinch off and moves within the cytoplasm. Endocytosis can be classed as either phagocytosis, which is what we can see here on the left, or it could be penocytosis when it is a liquid being taken in. So that's the difference. Phagocytosis is when it's surrounding and pinching inwards with a solid particle. The penocytosis is when it is a liquid. This requires energy from ATP for the cell to engulf and change shape around the material. Exocytosis is our last type, and this is the bulk transport of molecules out of a cell. So vesicles within the cell will move towards and fuse with the cell membrane. And as it fuses, it releases its contents outside of the cell. This process requires energy because ATP is needed to move the vesicle along the cytoskeleton up to that cell surface membrane where it fuses. Now, the surface area to volume ratio is key in considering how fast these types of transport can happen across the cell surface membrane. So exchange surfaces in organisms have very similar adaptations to increase this rate of transport. And surface area to volume ratio is one adaptation that we see. The relationship between the size of an organism or structure, so for example an organelle, and its surface area to volume ratio plays a significant role in the adaptations. So if we think about, first of all, what surface area to volume ratio means, just looking at cubes. If we were to work out the surface area of this cube, it'd be the area of one side, so 1 times 1 is 1, but there are 6 sides, so we times it by 6. That's why the surface area is 6 centimetres cubed. The volume is length times height times width, which would be 1 times 1 times 1. So the volume is 1. So our surface area to volume ratio, you would do the surface area divided by the volume. So 6 divided by 1 gives you a surface area to volume ratio of 6. You could do the same thing for these cells here as well. So on this one, we've now got a 2 centimetre in dimension. The picture actually doesn't show the full cube, I've realised, because you've just got the first layer where it should be 2 across. It is 2 up, but it's not going 2 back. And the same here, we've got 3 across, 3 up, but it's not showing you 3 back. But it'd be the same idea. You'd work out the surface area of one side and then times it by 6 for a cube. And then the volume is 2 times 2 times 2. You then do your surface area divided by your volume. And the pattern that we can see is the larger the object, in this case a cube, the smaller the surface area to volume ratio. And that would mean transport would happen slower because you have a lower surface area compared to the volume. So the smaller a structure is, the larger the surface area to volume ratio, and therefore the more rapid the transport across the membrane. So that's why small organisms, such as amoeba, they have a very large surface area to volume ratio. And that means they don't have any special exchange surfaces like lungs, because they can just do simple diffusion across their surface to meet their respiratory needs. But larger organisms, they will have a larger surface area to volume ratio, and therefore they will need adaptations to make sure they are getting enough oxygen in and enough carbon dioxide out, as well as it could be other structures within the body. And that's why we then start to see these adaptations. Not only that, the larger an organism, they will have more cells and therefore a higher metabolic rate. And they'll have a higher demand for oxygen, but also they'll be producing more waste products that need to be removed. So some of those key adaptations will come up in later topics, but it's ideas such as having microvilli on cells or having millions of alveoli in the lungs as well. So topic 5 then is mitotic cell cycle, and it's split into 2 parts, and we're beginning with replication and division of nuclei and the cells. So let's have a look at chromosomes to begin with. In eukaryotic organisms, the DNA is stored as chromosomes, and humans have 23 pairs of chromosomes, which means there's 46 in total. And you have 2 copies of every chromosome, and those matching pairs, or the 2 copies, are known as homologous pairs. And what we mean by homologous, wherever you see homo in biology, that means the same. So a homologous pair of chromosomes are 2 chromosomes that are the same size, and key is they have exactly the same genes, but they might have different alleles. And that's what we can see here on this diagram. We've got the gene for hair colour, but they've got different alleles. This chromosome has straight, this one has curly. And then you've got a selection of other genes. So this is one chromosome, this is another chromosome, because you count chromosomes by how many centromeres there are. So we do have 2 chromosomes in this image, and they are a homologous pair of chromosomes because they're the same size and they contain the same genes. A bit more then about chromosomes. In eukaryotic organisms, we said they're stored in the nucleus. And in order for that vast amount of DNA to fit in the nucleus, in this linear shape, that's how we describe the shape of the chromosome, it has to be tightly coiled up to fit. And to assist with that, the DNA wraps around proteins called histones. And I always say to think of this like if you were to try and pack away your hairdryer, if you wrap the cord around the hairdryer handle, it tightly compacts really neatly. And that's the same idea with DNA wrapping around histone proteins. It helps to really tightly compact and coil it up in a neat way so you're not going to damage the DNA. Chromosomes also have a centromere, which we can see here, and that is the pinching part in the middle of a chromosome. And after DNA replication, this is what a chromosome looks like. It's made up of two sister chromatids. Before DNA replication, it looks like a single stick-like structure, and it would still have a centromere in it. But at this point, the role of the centromere is to hold together those two sister chromatids. At the end of the chromosomes, you have what's known as a telomere, and that's a protective cap. And essentially, it's a DNA sequence and its proteins that are right at the end to protect the DNA during cell division. So it's these repetitive, short sequences that make the DNA longer, but those sections aren't coding. It is just to protect the coding regions of the DNA. And telomerase is an enzyme that adds bases to the telomeres to help maintain their length. So if you then go on to cell division, in eukaryotic cells, they enter the cell cycle and they can divide by either mitosis or meiosis. Prokaryotic cells divide by binary fission. And viruses do not undergo cell division as they are non-living. So we're going to focus on just mitosis in this video. The cell cycle is all of the stages involved in creating new cells. And it's split into three stages. You have interphase, which is the longest stage by a long way, and it's shown here in grey. So you can see the majority of the cell cycle is interphase, and that is split into G1, S phase, and G2, and we'll go through what those are in a minute. You then have M, or the nuclear division, which can either be mitosis or meiosis. We're just focusing on mitosis in this video. And then the last stage of the cell cycle is cytokinesis. So these are the three steps. Let's go through each of those in a bit more detail. So interphase, as I said, is the longest stage. G1 is when protein synthesis occurs to make proteins involved in synthesizing organelles. And then all the organelles can replicate. Because if you're going to split the cell in half, you need to make sure both of those new cells that are created have the correct number of organelles. So that's why we have to replicate all the organelles first. The cell is also checked that it's the correct size, has the correct nutrients, growth factors, and that there's no damage to the DNA. And if a cell doesn't pass those checks, then interphase will not continue. S phase is next, and that is the stage where DNA replication occurs. And then the last part of interphase is G2, and this is when the cell continues to grow, so that when it does split, the two cells are the same size as the original cell. It also has energy stores that are increased, and the newly replicated DNA is checked for any copying errors. And if any errors are found, those are either corrected or the cell is destroyed to make sure that these mutations aren't going to be replicated in multiple cells. Stem cells is the next part of this topic. And the definition of a stem cell is that it is an undifferentiated cell that can self-renew, which means to continually divide, and it can become specialized. And there are different levels of potency, which means their ability to divide into a number of different types of cells. So totipotent is the most potent, meaning it can differentiate into any type of cell. Then as we go down the list, it becomes less potent, so pluripotent, multipotent, and unipotent. So let's take a look at those terms in a bit more detail. So totipotent stem cells are the cells that can divide and produce any type of cell in the body. And during development, totipotent cells translate only part of the DNA, resulting in cell specialization. Totipotent cells occur only for a limited time in the early mammalian embryos. Pluripotent cells, those are also found in the embryos and can become almost any type of cell. They just can't become the placenta. For this reason, these are the ones that are used for research with the prospect of using them to treat human diseases, because you could potentially use these to create any type of cell or tissue except for the placenta. There are issues with this though, as sometimes the treatment doesn't work, or because stem cells continually divide, they can result in the formation of tumours. Additionally, there is the ethical debate on whether it's right to, first of all, make a therapeutic clone of yourself, because to use these types of stem cells in treating a patient, there would have to be identical DNA to the patient so they don't get rejected. And the only way to do that would be to make a therapeutic clone of themselves. You'd also have to create an embryo, and the embryo then gets destroyed, and that can be seen as playing God or murder. Now, in reality, they have actually got advances on this now, and they use something called induced pluripotent stem cells, but that isn't part of the specification. Next, then we go on to multipotent and unipotent stem cells, and these are found in mature mammals, and they can only divide into a limited number of different types. So multipotent stem cells is, for example, the stem cells that you get in bone marrow, and those can only specialize into or differentiate into the types of blood cells. So it's a limited type of different cells they can differentiate into. Unipotent means that it can only differentiate into the same type of cell. So, for example, skin cells can replicate and differentiate into more skin cells. So some potential uses of stem cells then could be in research and also in medicine. So for example, they could be used to repair damaged tissues or the treatment of neurological conditions such as Alzheimer's or Parkinson's, where there's damage to the neurons in the brain. Or it could be to look and use these stem cells in developmental biology, so for research purposes. Now, we did mention that stem cells, in theory, they could be amazing for treating different diseases, but because they self-renew, they could lead to the formation of tumours. And that takes us on to this concept here of what is a tumour and what is cancer. So mitosis, which we're going to be going on to, is a gene-controlled process. And there are genes that produce proteins that help to initiate and to control when mitosis stops, to make sure that new cells are only being replicated when the body needs those new cells. But mutations can occur in these genes that control mitosis, and if a mutation occurs in those genes, the proteins that are produced that are meant to control when mitosis starts and stops may not function, and as a result you have uncontrolled mitosis. And if mitosis is happening uncontrollably, that means you're going to be making new cells when the body didn't need those new cells. And that is what a tumour is. Now, not all tumours are cancerous. If it's cancerous, we call it a malignant tumour. And some properties of malignant tumours compared to the non-cancerous, which are known as benign tumours, is that a malignant tumour has the ability for some of those cells to break off and then spread in the blood, or it could be in the lymphatic system, and then lodge into new tissues in the body and create a secondary tumour. And we call that metastasis. They also can develop their own blood supply. And if they've got their own blood supply, that means they'll have lots of oxygen and glucose being supplied to these cells so they can respire aerobically more rapidly, produce more ATP, and that will mean mitosis can happen even faster. So many cancer treatments, for example, chemotherapy, work by preventing these rapidly dividing cells from entering mitosis. And therefore, that means the tumour can't get any bigger. And the way this works is some of those drugs using chemotherapy will target spindle fibre formation in metaphase, and this prevents mitosis from happening. And we'll have a look at how when we get to the actual stages of mitosis later on in this video. But ultimately, that prevents the cancer cells from dividing further. The downside is, though, it doesn't just affect cancer cells. It affects any fast dividing cells. So that is cells in your body such as hair cells, skin cells, the cells lining your intestine. Those are all fast dividing cells. And this chemotherapy will therefore affect all of those. And that is why chemotherapy has so many really unpleasant and nasty side effects. The next part of topic 5 then is looking at how the chromosomes behave in mitosis. Or in other words, we're going to be going through the stages of mitosis. So mitosis is one of the types of nuclear division that occurs in the cell cycle, and it creates two identical diploid cells. And diploid means you have two copies of every chromosome. It's used for growth, tissue repair, and it's also used in asexual reproduction in plants and animals and fungi. There are four key stages. You have prophase, metaphase, anaphase, and telophase. So you still have the cell cycle occurring. So you start with interphase, but then when you enter mitosis, you go through these four stages. And then at the end of the cell cycle, we have cytokinesis. So the way that people often remember this is just thinking about PMAT as an abbreviation, or is it an acronym? I don't know, PMAT, that's the way to remember it. Prophase, metaphase, anaphase, and telophase. So I'm going to talk you through what happens at each stage, and going back to what we said here, how the chromosomes are behaving at each stage. That's the phrase that is used to describe the position of the chromosomes. So prophase is the first step. And this is when chromosomes condense, meaning they coil up really, really tightly, and that means they become visible as individual chromosomes. And in animal cells, the centrioles, which are going to be responsible for releasing the spindle fibre, those are normally together at one pole of the cell, and instead they'll start to move apart to the opposite ends or the opposite poles of the cell. The centrioles that create the spindle fibres are released from both poles to start to create this spindle apparatus, and these will then attach to the centromere and the chromatids in the later stages. Plants also have a spindle apparatus, but they lack those centrioles. The next stage is metaphase. And in metaphase, the chromosome's behaviour is that they will line up in single file at the equator of the cell. And the equator is the term we use for the middle of the cell. Because it's a sphere, we use the term equator a bit like we do for the Earth. So they align in the centre, which is the equator of the cell. And at this point, those spindle fibres that have been released from the centrioles will attach to the centromere and some of the chromatids on those chromosomes. And this again helps them to align at the equator of the cell. The spindle assembly checkpoint also occurs at this stage, and this is where there's a check to ensure that every chromosome has attached to a spindle fibre before mitosis can proceed into the next stage, which is anaphase. So in anaphase, this is when those spindle fibres start to shorten and move back towards the centrioles. And as they do that, it pulls the centromere and chromatids and it splits that centromere in half. And that is what results in the chromatids, those sister chromatids, being separated and being pulled towards the opposite poles of the cell. So that's what we've got written just here. Now, this stage does require energy in the form of ATP, which is provided by aerobic respiration in the mitochondria. So this links back to what we were saying in terms of cancer, the fact that malignant tumours have their own blood supply, therefore they get more oxygen and more glucose for aerobic respiration to happen at a faster rate. Therefore, there's more ATP, and that is going to be helping in this stage here, anaphase. But when we talked about the drugs, chemotherapy, affecting that spindle fibre formation, if spindle fibres aren't forming, you're not going to be separating the chromatids, the opposite poles, and therefore you don't end up with two new cells that have the correct number of chromosomes, and those cells would therefore be destroyed. Now, lastly, we've got telophase, and this is when the chromosomes are now at each pole of the cell, and the chromosomes start to become uncondensed. So they get longer and thinner again, and therefore they are starting to become no longer visible as individual chromosomes. The spindle fibre will also start to disintegrate at this point, and we can see that nuclear membrane starts to reform. The final step is the cell cycle. This doesn't count as mitosis, but cytokinesis is the last part of the cell cycle, and this is when the cytoplasm splits into two genetically identical cells. So in animals, you get this cleavage furrow forming in the middle of the cell, and the cytoskeleton causes the cell membrane to draw inwards, and that is then what causes the cell to split in two eventually. In plant cells, the cell membrane splits into two new cells due to the fusing of vesicles from the Golgi apparatus, and then The cell wall forms new sections around the membrane to complete the division into two cells. Now, you could be asked to observe mitosis under a microscope. And often this is done with root tips of plants such as onion and garlic. And that is because at the very tip of those roots, and that's because that is where lots of mitosis is occurring, and therefore you're more likely to be able to observe mitosis at those points. So a thin slice of those root tip is placed on a microscope slide, broken down with a needle. You'd also often add acid and heat so that you are breaking down the connections between the cell walls. So you get this thin layer when you squash down on that sample. You also add a stain, and the heat helps that stain to really stain the chromosomes so they become visible. So that's what we've got here. A stain is added to make the chromosome visible, and the cover slip is then pushed down on. The pushing down is going to squash that tip so you get a single layer of cells so that the light can pass through, and therefore you can observe what is going on in those cells. And you might get something that looks a bit like this. Now, all of these ones here, where it just looks like a pinky purple circle, you can't see individual chromosomes in those. And that would mean those cells are in interphase of the cell cycle. And most cells will look like that because interphase is the longest stage of the cell cycle, and therefore most cells will be in interphase. But in these two, we can see the chromosomes are visible, and it looks like at this point they are lining up along the equator. So those two cells would be in metaphase. And you can work out what's known as the mitotic index, and this is by counting how many cells are visible in your field of view and how many cells are visible that are in the stage of mitosis. And you would then use the formula, the number of cells in mitosis divided by the total number of cells, to give you a mitotic index, which is essentially an index to give you an idea of the rate or how much mitosis is occurring. So you could do a comparison between different tissues. So here is topic 6, and in this topic we're going to be looking at nucleic acids and protein synthesis, starting with 6.1, which is the structure of nucleic acids and replication of DNA. DNA and RNA both occur in the monomer form and as the polymer form, and the monomers of DNA and RNA are known as nucleotides. And the nucleotide consists of a nitrogenous base, which for DNA can be either cytosine, thymine, adenine or guanine, and for RNA it could be uracil instead of thymine, cytosine, adenine and guanine. Both of them then have a pentose sugar, and for DNA that is deoxyribose, and for RNA it's ribose, that's why there's the D and the R difference, and then they both have one phosphate group. Now with these bases, the nitrogenous bases, you can categorise them according to their structure. Adenine and guanine are classified as purines, and that means that the nitrogenous base is made up of 2 rings, which we can see one ring here and a second here, and that's the case for both adenine and guanine. Then we have cytosine, thymine if it's DNA, uracil if it's RNA. Those 3 nitrogenous bases are pyrimidines, which means that they only have 1 ring structure. So these bases have a complementary base, which means when you have the 2 DNA strands, the bases will align opposite a particular base, which is complementary to them. And thymine and adenine are complementary, or if it's RNA, it would be uracil and adenine are complementary, so they'll always align opposite each other. And guanine and cytosine are complementary to each other, so they'll always align opposite. And what that means is you always have a purine and a pyrimidine base facing each other. And this complementary base pairing is really important because when the DNA replicates, it ensures that you get identical copies being made. Both DNA and RNA nucleotides, as I said earlier, can form a polymer, and the way that you go from having a monomer to having a polymer is a condensation reaction. And that is when water is removed, joining together 2 molecules, and a bond is formed. And the bond that forms is a phosphodiester bond. The polymer of the nucleotides is known as a polynucleotide, and that phosphodiester bond that forms is a really strong covalent bond. You end up getting what is known as a sugar phosphate backbone, which is where you have the sugar, which is here, the sugar, the phosphate, the sugar, the phosphate, the sugar, the phosphate, all attaching to each other, although that's actually showing ATP here, pointing out that you need energy for this reaction. So I'll do it on this one. We've got the sugar, the phosphate, the sugar, the phosphate. That's your sugar phosphate backbone, and it's all connected via these strong covalent bonds, the phosphodiester bonds. So you end up with a really strong polymer, which is important because you do not want that genetic code to be changed, such as mutations, because that is your hard copy of all of your genetic material. Next up, we have a look at ATP. This is a nucleotide derivative, so it's very similar in structure. The key difference is it contains 3 phosphate groups, so these 3 phosphate ions. You still have a sugar and a nitrogenous base, but it is always adenine and it is ribose. So you have adenine, ribose, and 3 phosphate groups. And ATP is essential for metabolism. It is an immediate source of energy. When you break these bonds between the phosphate groups, it releases energy. And that can be used for all the different chemical reactions, which is what metabolism is, all the chemical reactions in the cells, and also some processes. So the way this happens then is, first of all, ATP is made during respiration via condensation reaction, and it uses the enzyme ATP synthase. So we have ADP, which is diphosphate, which is when you have 2 phosphate groups, and you need to add on that inorganic phosphate. That's what PI is. So that inorganic phosphate group gets added on via a condensation reaction, and that's how you make ATP. And because it was a condensation reaction, water is removed. Now, to release the energy from ATP that's stored in those chemical bonds, you have to hydrolyze the ATP, and that is done using the enzyme ATP hydrolase. So this time it'd be a hydrolysis reaction, which is when you are bond to separate 2 molecules through the addition of water. So we're going to break the bond between the phosphate groups. That releases energy, and that then means we have ADP and PI. So breaking one of those bonds between the phosphate groups releases a small amount of energy. That means you don't have wastage energy. So if you need more energy, you would just break the bond between more ATP molecules. And that can then be used in chemical reactions. That inorganic phosphate group can also be added onto other molecules, and that's known as phosphorylation, when you add a phosphate group onto another molecule. And when you do that, it makes the new molecule more reactive and it gains energy. And that's actually what you see happening in the first stage of respiration in glycolysis. So if we have a look at DNA in a bit more detail, DNA, or deoxyribonucleic acid, that codes for the sequence of amino acids in the primary structure of a protein, which in turn is what determines that 3D structure and function of a protein, because it determines where the bonds can occur and therefore how it folds in that 3D shape. It's essential that the cells contain a copy of this genetic code, and it's really essential it gets passed on to new cells without being damaged, because if it does get damaged, you will change your DNA base sequence, therefore code for a different primary structure, potentially, and you end up with a non-functioning protein. DNA, as we said earlier, is a polymer, and the polymer is made up of 2 chains, which will then form a double helix. The nucleotide we talked about previously, you've got your phosphate group, deoxyribose, and one of those 4 nitrogenous bases. So the polynucleotides we also briefly talked about, so that is the polymer. Created by those condensation reactions, you get those phosphodiester bonds, which are the strong covalent bonds. The sugar phosphate backbone, you can see a little bit clearer now because it's shown in blue around the outside of this double helix. And the DNA polymer occurs in pairs joined together by hydrogen bonds. So these coloured parts sticking out here are representing the complementary bases, and at every point that would be either 2 or 3 hydrogen bonds holding together those 2 chains. The hydrogen bonds can only form between the complementary base pairs, so you'll have those hydrogen bonds between guanine and cytosine, and adenine and thymine. And as I said, you either have 2 or 3. Adenine and thymine can form 2 hydrogen bonds, whereas cytosine and guanine form 3 hydrogen bonds. So why are these complementary bases really important then? The 2 strands of the DNA molecules, first of all, are anti-parallel. And what we mean by that is they run in opposite directions. So on this side, you would have, this is the sugar phosphate backbone, so that would be your phosphate and the sugar at the bottom. Whereas this side, it's upside down. You have the sugar and the phosphate on the top. You can't quite see it very clearly in this picture, but that is the concept of anti-parallel. And instead of saying top and bottom, you have it described as 5' and 3' end, which is referring to which carbon within the sugar ring is most exposed. So you'd have a 3' end and a 5' end, and then in anti-parallel it would be the opposite way round, so you'd have 3' and 5'. Now, the complementary base pairing is important because it ensures the specificity of DNA replication and transcription processes. The 5 to 3' orientation of one strand will align with the 3 to 5 of the other one, and it allows the accurate copying and therefore the accurate expression of genetic information, which means that you have your correct DNA base sequence, which will then code for the correct amino acid sequence. Now, we talked about RNA as a nucleotide and as a polymer, and one of the polymers that RNA can form is messenger RNA, or mRNA for short. And mRNA is a copy of one gene. mRNA is made in the nucleus, so because it's a copy of one gene, it has to be copied in the nucleus because that is where the DNA is located. You get one copy of it, which we'll be looking at later on in this lesson in transcription, because that's how you make mRNA. It then leaves via the nuclear pore to the cytoplasm, where it will find a ribosome to attach to. So this is really important because DNA cannot leave the nucleus because it is so large, whereas mRNA is much shorter because it's only a copy of one gene. But this is actually a good thing. We don't want DNA to leave the nucleus because that is your hard copy of your genetic material. And if it got into the cytoplasm, there are enzymes in the cytoplasm that could break down that DNA. So instead, when the genetic code is required to make a protein, we make an mRNA copy of one gene. That can leave the nucleus because it's shorter. That can be used to create the protein. And if the mRNA breaks down, which it will do, that doesn't matter because you just make another copy of it from your DNA. So it is short-lived because it is going from the nucleus into the cytoplasm, where it will get broken down after it's been used in protein synthesis. And it is single-stranded. It forms a straight line, and every 3 bases on your mRNA sequence is known as a codon. So 3 bases, 3 consecutive bases on mRNA, which codes for one amino acid, a specific amino acid, is known as a codon. Next, we're going to have a look at how DNA replicates, and it replicates through semi-conservative replication. And what we mean by that is, each time the DNA replicates, one strand is conserved and one strand is newly synthesized. So it's semi-conservative, but it's really important that you say one strand, not half, because half could imply it all breaks apart and mixes up and half is new, half is old. So you have to specify one strand is new, one strand is old or conserved. It's in this stage that mutations could occur because sometimes copying errors do happen in DNA replication, and they are random, spontaneous, and that would result in a change to the DNA base sequence and therefore it would code for a different amino acid potentially. But we're going to come on to that right at the end of the video. So this happens in S phase of interphase of the cell cycle. And this concept of the 3' and 5' end are useful in describing the stages of DNA replication, because instead of saying the start or the end, you can talk about the 3' or 5' end. And this is relevant because we're going to look at the enzymes involved in DNA replication, and the enzyme that catalyzes one of the stages of DNA replication is complementary in shape, so the active site is complementary in shape to the shape of DNA only at the 3' end. And that means that that enzyme can only attach at the 3' end of the DNA. So describing the stages of DNA replication could be a potential long answer question. There's 4 key steps. Step 1 is the DNA, first of all, has to be separated apart from that double-stranded double helix. And that is done by the enzyme DNA helicase. DNA helicase breaks the hydrogen bonds between the complementary base pairs, and that is what causes the double strands to unzip, unwind, and you get these replication forks, which is what we can see here. It's actually labelled slightly differently on the diagram, but there's our DNA helicase breaking those hydrogen bonds, and you get one strand over this way, one strand over that, and that is known as a replication fork. Step 2 then, those 2 separated DNA strands will now both act as a template for new DNA strands in DNA replication. Free-floating DNA nucleotides that are always within the nucleus will align opposite their complementary bases, and they'll form hydrogen bonds between those complementary bases. But at this point, the nucleotides aren't joined together with that phosphodiester bond. And that is where the enzyme DNA polymerase comes in. DNA polymerase is able to join together those adjacent DNA nucleotides through the catalyzation of the condensation reaction, and it forms the phosphodiester bonds. And that then results in two daughter DNA molecules, each consisting of one of the original parental strands and one newly synthesized strand. So one key focus is knowing the process of DNA replication, but you also need to know the roles of different enzymes involved. And we've talked about DNA polymerase. We said that DNA polymerase catalyzes the formation of the phosphodiester bond between those adjacent nucleotides on the newly synthesized chain. And this is the enzyme where I was saying it is complementary in shape to the 3' end. So DNA polymerase will always bind to the original template strand at the 3' end. That means the newly synthesized strand, which is anti-parallel, will have new nucleotides being added in the 5' to 3' direction. And that leads to differences between the leading strand and the lagging strand in DNA replication, which if I actually just go back to this diagram here, we can start to see that concept of leading strand versus lagging strand. So the leading strand is where you do have this continuous addition of the bases being catalyzed with that condensation reaction. The lagging strand, you get it in separated sections, which we call Okazaki fragments. And that's where we see the role of DNA ligase. So DNA ligase is involved on the lagging strand during DNA replication. So DNA polymerase synthesizes short DNA fragments called Okazaki fragments in that opposite direction of the replication fork movement. And as a result, those sections, those Okazaki fragments, have to be joined together. And that is what DNA ligase does. It joins those Okazaki fragments by catalyzing the formation of phosphodiester bonds, completing the synthesis of that lagging strand on the DNA molecule. Next, then, we have a look at protein synthesis. So for protein synthesis, the first concept is you need to have an understanding of the genetic code. And there's 3 special features. The genetic code is degenerate, it's universal, and it's non-overlapping. What we mean by degenerate is amino acids. There are 20 amino acids, but there's actually 64 triplets of bases. So amino acids can be coded for by more than one triplet of bases. Universal is that the same triplet of bases codes for the same amino acid in all organisms. And lastly, non-overlapping. This is when each base in a gene is only a part of one triplet of bases, or you could describe that as a discrete unit. So each codon is its own separate discrete unit. There's no overlapping of the bases in multiple triplets. So the advantage of this is the fact that the genetic code is degenerate means even if a mutation occurred, changing one of the bases in a triplet, that new triplet due to the mutation might still code for the same amino acid. So it reduces the impact that mutations might have. The fact that it's universal has been really helpful in genetic engineering because it means that we can take a human gene and insert into the DNA of bacteria, and that bacteria will then produce the human protein. Non-overlapping is also linked to the concept of mutations. So it's really useful that each base is only part of one triplet, because if a mutation did occur in a triplet, then at least it's only one codon which is affected and therefore only one amino acid. So protein synthesis occurs on the ribosomes or the rough endoplasmic reticulum because they're rough because they have ribosomes. Transcription actually happens in the nucleus, and that is the first stage, and that is where the DNA is copied into an mRNA sequence, and that's just for one gene. Translation is the part that happens on the ribosome, and this is where the polypeptide chain is created. So we're going to go through those 2 processes, but just a few other key things that you need to know. Number 1 is what introns and exons are. So introns are sequences of bases on the DNA, so in a gene in particular, that do not code for amino acids and therefore the polypeptide chain. So these introns have to be cut out or removed, or spliced is a technical term, out of the mRNA once the copy of the gene has been made. So mRNA is made and then the introns are spliced out. That then just leaves the exons behind, and exons are sequences of bases in the gene that code for sequences of amino acids. So that's the coding section that's needed to code for the polypeptide chain. There's also start and stop codons. So at the start of every gene, there is a start codon, which is 3 bases, which will code for a particular amino acid, which will initiate translation. The stop codon is the final 3 bases at the end of every gene, and these will not actually code for any amino acid, and as a result it causes the ribosome to detach, and therefore it stops or ends translation. So let's go through the 2 stages then. Transcription is the first step, and this is the process that's happening within the nucleus, where we get an mRNA copy of one gene on the DNA being created. So the steps are very, very similar to DNA replication. First of all, DNA helicase breaks the hydrogen bonds between the bases in the 2 DNA strands to unwind that double helix and to separate the 2 strands. Here's the first difference though. Only one of those strands will act as a template, whereas in DNA replication, both of those strands of DNA will act as a template. Next then we have 3 mRNA nucleotides lining opposite the exposed complementary bases on the DNA. Same concept, but this time it's mRNA nucleotides instead of DNA. And then the next bit again is very similar, it's just a slightly different enzyme. This time it's RNA polymerase that will join together the adjacent nucleotides, forming a phosphodiester bond through that condensation reaction. And that is how you then get an mRNA polymer. And once that one gene is copied, then you would have the introns spliced out, and then the mRNA can leave the nucleus via the nuclear pores and go and attach to a ribosome in the cytoplasm. And that then takes us on to the next step, which is translation. So once that modified mRNA has left the nucleus via the nuclear pore, it attaches to the small subunit of the ribosome at the start codon. tRNA molecules, which are a different type of mRNA that carry specific amino acids, they have anticodons on them, which are 3 bases complementary to a codon on mRNA. So the tRNA molecule with the complementary anticodon to a codon will align opposites, and the ribosome is able to hold 2 tRNA molecules at a time. The 2 amino acids that will be at the top of the tRNA molecule are therefore delivered, and it is specific based on the anticodon and the codon, and they are then able to join via a peptide bond, and this requires an enzyme, and it also requires ATP, energy released from ATP, to form that peptide bond. The tRNA is then released, and the ribosome will move along the mRNA molecule to the next codon, and that process happens again. So the next tRNA molecule can then align opposite its complementary codon compared to its anticodon, bringing a specific amino acid. And this process continues until the ribosome reaches the stop codon at the end of the mRNA molecule, and that is what causes the ribosome to detach and therefore ends translation. That then results in your finished polypeptide chain, which has been created, which can then enter the Golgi body for further folding or modification, such as maybe adding a carbohydrate to it. So the last concept in this module is gene mutations. And a gene mutation is a change in the base sequence of DNA that might result in a different polypeptide. They occur randomly and spontaneously during S phase of interphase, which is when DNA replication happens. But these mutations are more likely to occur if you're exposed to certain chemicals or high energy radiation. So for example, UV light, gamma rays, X-rays, and the chemicals we call carcinogens, so mustard gas, for example. A gene mutation could result in either a base being deleted or substituted. So for example, we've got our original DNA base sequence here, and we can see that base cytosine has been substituted, so changed, for the base adenine. So that is a substitution gene mutation. The next one we've got is a base has been added or inserted. It's exactly the same sequence, except thymine has been added at this point. So that means we've now changed all of the subsequent triplets after that mutation, and we've also got an extra base just not a part of any triplet. We call that a frame shift when it shifts along like that. Deletion has a similar effect. If you delete one of the bases, all of the triplets after the point of the mutation change, and that means it's a frame shift. This time, though, it's all of the triplets move back one position. So this one, we don't even have a triplet anymore, so that wouldn't even code for an amino acid. So you're coding for different amino acids, and you'd have one fewer. So sometimes a substitution can be described as silent, meaning that the new codon still codes for the same amino acid. And that's because of that feature of the genetic code we talked about, it being degenerate, meaning that multiple triplets of bases can still code for the same amino acid. However, those deletions or insertions, those can result in frame shifts where after the point of mutation, all of the subsequent codons are changed and therefore will probably code for different amino acids, and you'll get a very different primary structure or sequence of amino acids. Therefore, the protein will fold into a very different shape and it will be non-functioning, or it might have a completely different function. In terms of enzymes, that would mean the active site would be completely changed and the enzyme would no longer catalyse the reaction it's meant to. So that is far more harmful.
Topic 7 then, we're going to be looking at transport in plants, starting with the structure of transport tissues. So this table here summarises the distribution of xylem and phloem across the different parts of the plant. So looking at the distribution in stems, roots and leaves. So within the stem, the xylem tissue is located towards the centre of the stem and it forms a central vascular bundle. It's surrounded by the phloem tissue, and the xylem vessels are arranged in a characteristic pattern, often forming a ring or several rings within the stem. Whereas the phloem is found between the xylem, and as we said just here on the outside, and it's the outermost layer of the stem and it forms a ring around those xylem. In the roots, the xylem tissue is typically located in the centre again, forming that vascular bundle, and the xylem vessels radiate outwards, with the youngest xylem tissues towards the centre and the oldest towards the outside or the periphery. The phloem is arranged in a ring surrounding that xylem, and this arrangement allows for the efficient transport of sugars and other organic compounds produced in the leaves to the roots for storage. Finally then, in the leaves, the xylem are found in the midrib and the veins, and it extends the leaf lamina, and it provides structural support and it facilitates the transport of those water and mineral ions that have been absorbed by the root hair cells to the leaves. The phloem are located adjacent to the xylem in the veins, and it transports organic compounds such as sugars produced during photosynthesis from the leaves to other parts of the plant. So if we have a look at the structure of phloem and xylem, here we can see the phloem tissue, and it's made up of two key types of cells. We have the sieve tube element cells, which are shown here in the middle, and the companion cells, which are on the outside or either side of the sieve tube element. The sieve tube elements were initially living with all the organelles, and they are still classed as living cells, but as they start to develop, they then contain very few organelles and no nucleus. And you also have these perforated end walls, which makes it like a sieve, hence the name sieve tube elements. And this enables the sugars, the organic substances, to move up and down through the sieve tube element with little resistance from those organelles. Now, because it doesn't have organelles though, it is reliant on the companion cells to provide ATP for the active transport of organic substances. So companion cells do have all their organelles, and they have lots of mitochondria for aerobic respiration to be able to provide lots of ATP. Next, then, if we have a look at the xylem tissue, xylem vessels are composed of elongated hollow cells called vessel elements. This is actually looking through a cross section going into one of those, and in red here this is showing you one of those hollow xylem vessels. Now these cells are dead when they're mature, and they have lignified cell walls, so there's lots of lignin in them, which makes them waterproof and really tough. As you go through the tube as well, there are perforations called pits or pores. The structure of the xylem vessel elements allows for the efficient of a unidirectional flow of water and mineral from the roots to the shoots through capillary action and cohesion tension mechanisms, because it essentially makes one continuous long hollow tube so water can flow through it as one continuous column of water. Next then we have a look at the transport mechanisms. So we're going to be looking at the transport of water through a plant first of all. And water enters a plant at the root hair cells, and that is by osmosis. And root hair cells are adapted to maximise osmosis by having thin walls, so there's that short diffusion distance, because osmosis is a type of diffusion. And also they have that long protruding part that creates a large surface area to maximise the absorption of water by osmosis as well. Once the water is then inside of the root hair cell, it's then going to travel to the xylem either through what we call the symplast pathway or the apoplast pathway, which we can see here in the diagram. The symplast is shown in blue and the apoplastic pathway is shown in orange. So the apoplast pathway is through the cell walls, which we can see here. The water moves through those cell walls. The water can enter the cell wall and move due to cohesive forces, meaning the water sticks together with hydrogen bonds. And it forms this continuous stream of water which moves through the cell walls towards the xylem. This pathway transports the water really quickly as there's little resistance to the water in the cell wall, so it can move through quickly. However, when it gets to the endodermis, there are a specialised layer surrounding the vascular tissue, which is known as the Casparian strip, and that's composed of hydrophobic or waterproof suberin, and that forms this waterproof impermeable barrier in the cell walls. And at that point, it forces the water and all the dissolved solutes within it to enter the symplast pathway. Then if we have a look at the symplast pathway, this is when the water and the solutes cross the endodermis through the plasmodesmata, which is a gap here in the cell walls. The symplast pathway is moving through, or it's when the water is moving through the cytoplasm of the cell. So the water moves through the cytoplasm, and when it gets to a cell wall, it has to pass through a plasmodesmata, which is one of these gaps. Each successive cell cytoplasm has a lower water potential than the next, and that's how the water is moving. So it moves through the cytoplasm by osmosis, and this is a slower process compared to the apoplast pathway. You need to know some adaptations that plants have linked to the idea of transpiration or reducing transpiration and reducing the water that can be lost from a plant. So plants have stomata, and stomata we're going to be looking at in more detail later on in this video. They're the tiny pores on leaves that can open and close to reduce water loss, but open to enable gases to exchange. So xerophytes are plants that have adaptations to reduce water loss because these plants are found in environments with very, very little water, or it could be water but the high salt concentration, or it could be very, very hot as well. But traditionally we mean there's limited water. Marron grass is an example of a xerophyte, and it's found at the sand dunes, which despite it being next to the ocean, which is water, there is a limited water supply due to the fact that sand is so porous, so the water runs away. And here we can see a microscope image looking at the cross section through a leaf of a xerophyte, so we can see some of the adaptations that they have to reduce water loss. First of all, we can see that the leaf is curled up, and that curling up means that any water that does evaporate out gets trapped in this area, which increases the local humidity, and therefore it reduces the water potential gradient from the inside of the cells to the outside, and therefore there's less transpiration. There are also these hairs which have the same advantage in that they trap the moisture in the air, and that increases the local humidity, therefore decreasing the water potential gradient, so there's less transpiration. The sunken stomata is yet again creating that increased local humidity because it's lower down in the cell, so as the water evaporates, again you get this trapped humid air. On the outside here there's a thicker cuticle which helps to reduce the water loss by evaporation of the surface. And also not shown in this image, but there are longer root networks so that more water can be reached to absorb more water into the plant in the first place. So that then leads us on to what is transpiration and how does the plant transport water through the xylem? So we've looked at how the water gets into the roots, how it gets from the roots to the xylem. Now we need to see how it transports through the xylem through the rest of the plant and how it can actually leave the plant. So transpiration is the evaporation of water from the internal surfaces of the leaves, and it's followed by the diffusion of water vapour through the stomata. And the stomata, as we said, is these holes in the leaves which are created by guard cells. And plants can control water loss using these guard cells. The guard cells will swell and bend when the plant has a lot of water in them. And when they swell and bend, that ends up in creating this opening, which is known as the stomata. Now that enables carbon dioxide to diffuse into the leaf, but at the same time, water vapour is able to evaporate out. So the stomata open when it is light and the plant cells are turgid. You can actually measure the rate of transpiration with a piece of apparatus called a potometer as well. So thinking about how this water moves up the xylem then. We've just said that transpiration is evaporation through those stomata on the leaves. And as evaporation is the conversion of liquid water to water vapour, there are certain factors that are going to increase the rate of that. Temperature is one of them, because as you increase the temperature, the water molecules gain more kinetic energy and therefore they're moving more rapidly and you're going to have more evaporation. If the surrounding area is less humid or there's a lot of wind to carry away evaporated water molecules, then you have a steeper water potential gradient and therefore more water is going to evaporate out. We've partly touched on this concept here as well already, but it's just pointing out the starting place of this water. So water is absorbed by osmosis via the root hair cells, and there's a big root network to increase the surface area to maximise osmosis. Once that water then reaches the xylem, we're going to be looking at how it transports through those hollow tubes that we talked about that make up the structure of those xylem vessels. So the water can move in one direction only through the xylem, which is typically against gravity, so going upwards, and that's due to the transpiring water from the leaves, which creates this pulling force from where the water is being lost. This is often known as transpirational pull or transpiration pull. Water is absorbed by the root hair cells by osmosis, and that water then moves up the xylem within the stem, or in this picture it's up the trunk, and you get this continuous water column. And the reason for that is water molecules stick together by cohesion. So water is a polar molecule, which means you've got a slight positive charge on the hydrogen atoms and a slight negative charge on the oxygen atoms. For that reason, hydrogen bonds can form between different water molecules, and those hydrogen bonds between them all creates this cohesion, this sticking together of water molecules to create this continuous column of water. Now, those water molecules can also adhere to, so stick to, the walls of the xylem, which is what this grey here is representing, the walls of the xylem. So you would also get hydrogen bonds sticking this water molecule to the walls. Now, this cohesion, as we said, results in that continuous column of water, also known as the transpiration stream, in the plant stem. Tension is put on that transpiration stream, or in other words, a pulling force, when water evaporates out of the stomata. And that's because you get this negative pressure. As the water evaporates out, there's a negative pressure, and that creates this tension or pulling on that continuous column of water. This movement of water out of the stomata results in the water column being pulled up the xylem towards the stomata, and that is known as the transpiration pull. This pull will draw up the water, and it also puts tension on the xylem because the water is stuck to or adhering to the xylem walls. So this pulls the xylem inwards, making the hollow tubes narrower and longer. And this change in diameter of the xylem is measurable. For example, the diameter of a tree trunk will change according to its transpiration rates. And when it is a narrower tube, it makes it even easier to pull that column of water up the xylem. Next then we're going to have a look at the mass transport of organic substances in plants. So these are, for example, sugars such as glucose or sucrose that are made from the photosynthesis of a plant. And those sugars are needed for respiration. So it's important that although the sugars, organic substances, will be made in the leaves, they can be transported up and down the phloem to any respiring cells in the plant. So this takes us back to what we were talking about in terms of the phloem tissue, which we mentioned earlier, those sieve tube elements and the companion cells, which are involved in this mass transport of the organic substances. Translocation is the term that we use to explain how organic substances are transported in a plant. And this is an active process, meaning it requires energy. So we're going to go through this mass flow or translocation from what we describe as the source, which is where these organic substances are produced, which is the leaves and photosynthesis, to the sink, which is what we call the cells which are going to be using the sugar. Now, although it says a root here in brackets, is any respiring cell. So we're going to look at how those organic substances are transported from the source, which is where they're made, to the sink, which is where they're used. First of all, we're going to have a look at it with this model here, this source to sink explanation. So we've got the phloem shown in grey, the xylem shown in green. And we did talk about the structure, that you have the xylem typically in the middle and the phloem on the outside. And we've got a tank of water here as well, just to represent this model. So we've got the source cell is the photosynthesizing cell, and we've got our sink cell is the respiring cell. The photosynthesizing cell, because it's going to be creating lots of sugars, that lowers the water potential of that cell. And as a result, water can enter by osmosis. At the respiring cell, you're using up lots of organic substances and sugars. So that means there's going to be a more positive water potential or less negative, and therefore water will leave those sink cells by osmosis. Now, this difference in whether the water is moving in or out by osmosis is going to affect the hydrostatic pressure. As water moves in at the source cell, there's an increased hydrostatic pressure, and as water is leaving at the sink cell, you have a decreased hydrostatic pressure. That then creates this pressure gradient, and as a result, the sugary solution in the source cell is going to be forced up and through the phloem towards the sink cell due to that high hydrostatic pressure in the source cell in comparison to the sink cell. So that's the concept of mass flow in terms of the pressure changes, but you also need to know how does the sucrose, which we're using as our example of the organic substance, get from the source cell into the sieve tube element in the first place. And this is where we see the role of active transport and the need for energy. So step one is we've got the photosynthesizing cell, such as your leaf cell, is going to be creating that organic substance sucrose. And that creates a high concentration of sucrose at the site of its production. And therefore it can diffuse down its concentration gradient. That would be via facilitated diffusion though, because it's too large to move through the plasma membrane. And it goes into the companion cell. At this point, there'll be active transport of protons or hydrogen ions from the companion cell into spaces within the cell wall. And because it's active transport, that requires energy. That creates a concentration gradient, and therefore the protons move down their concentration gradient via proteins into the sieve tube elements. And that is how we get the sucrose into the sieve tube element. Co-transport of sucrose with hydrogen ions occurs via protein co-transporters to transport it into those sieve tube elements. This is now where it links to that model we were talking about. Because we've got that sucrose inside of the sieve tube element, it lowers the water potential. That means water is going to enter by osmosis. That increases the hydrostatic pressure, and it forces the liquid to be moved towards the sink cells. At those sink cells, sucrose is going to be converted into glucose and used in respiration, or it might be stored as insoluble starch. More sucrose is actively transported into the sink cell, which causes the water potential to decrease. This results in osmosis of water from those sieve tube elements to the sink cell. So some water also returns to the xylem as well. The removal of that water decreases the volume in the sieve tube element, and therefore the hydrostatic pressure decreases. And there we have that pressure gradient then, because you've got a high hydrostatic pressure at the source compared to the sink end of the sieve tube element, and therefore the solution moves en masse.
So topic 8 for Cambridge International then, we're going to be looking at the circulatory system, transport of oxygen and carbon dioxide, and then finally the heart. So starting with the circulatory system, large animals require circulatory systems because they have high metabolic rates, which means they're doing lots of chemical reactions at a very fast rate, but they have a lower surface area to volume ratio. So they need to have these transport systems to ensure that they are getting the resources that they require for the chemical reactions, whilst also removing the waste products. So each animal has a circulatory system adapted to its particular needs, and all circulatory systems will transport gases, for example carbon dioxide and oxygen, as well as nutrients like glucose around the body within liquids such as the blood. And the liquid is transported around in vessels, and there's always a pump to help move the liquid. And one type we're going to look at is the double closed circulatory system. So in a double closed circulatory system, the double is referring to the fact that you've got two loops, and the closed means the blood always remains inside of a blood vessel. Now, for birds and most mammals, this is what they have, a double closed circulatory system. And one of the circuits is taking the blood from the heart to the lungs to become oxygenated and to remove the carbon dioxide. And the other circuit is delivering oxygenated blood to respiring cells around the rest of the body. There's quite a lot of details that you need to know about the different blood vessels, but because this is one of the summary videos, I've got it all summarised here in one table, so you can screenshot or pause and draw this down as your own version, so that you've got it in your notes, or you can make it as flashcards. But the key thing you need to know is about the arteries, arterioles, capillaries, venules and veins. Those are the 5 types of blood vessels that you need to know the structures and properties of. So we've got information here on the smooth muscle and the elastic layer, the collagen layer, the overall thickness of the wall, and whether they contain valves or not. And then I've got it linked to how that structural feature links to its function as well. So we can see that the arteries, they've got thicker muscular layer than the veins.
That enables this constriction and dilation of the blood vessels, so the lumen gets larger and wider, and in that way it can control the volume of blood. Arterioles, they've got a thicker muscular layer than in the arteries, and that's so they can contract with even more force to help restrict the blood flow into the capillaries. You don't have a muscular layer in the capillaries. You do in the venules and veins, relatively thin, so it can't actually control the blood flow. The elastic layer is a similar concept here in terms of the differences. So you've got a thicker layer in the arteries and veins, and it helps to maintain the blood pressure. It's what enables that stretch and recoil in response to the heartbeat. You've got a thinner layer in the arterioles as the pressure is getting lower the further away you are from the heart. No elastic layer in the capillaries or in the venules, and there's a very thin layer in the veins. Then we get to the collagen. Collagen is that outer layer to provide support, and you've got a thinner layer in the arterioles compared to the arteries, none in the capillaries, none in the venules, but you have a lot in the veins because it needs that structural support. Lastly then, if you consider all of those, you get the overall thickness of the walls. So the walls are thicker in the arteries and the veins, and that's one thing you can really notice on electron micrographs. They're thinner in the arterioles because the pressure is slightly lower. The capillaries, the reason they don't have muscle, elastic layer or collagen is they're only one layer thick. So you've only got one layer of cells making that epithelium of the capillaries. And that's to provide a really short diffusion distance because it's the capillaries where we have gas exchange happening and also tissue fluid formation. In the veins, it's very thin, and you can see that again in any microscope images. You can see just how thin the wall of a venule and then also the veins are compared to the arterioles and the arteries. And then lastly, valves. You only have these in the venules and veins, and that's to prevent the backflow of blood. There's lower pressure in the venules and veins, and that's why they need these valves to prevent the backflow. So here we can see the capillary in more detail, that single layer of cells making that endothelium, and they form capillary beds, so networks of capillaries where you have many of them branching, and that would be at exchange surfaces. So for example, surrounding the villi in the small intestines and surrounding the alveoli in the lungs. They have a narrow diameter, and the advantage of this is it slows down the blood flow because they're just wide enough for red blood cells to fit through. And because that then slows down the blood flow, that means there's more time for exchange, such as diffusion, to occur. So here we can then see some of those differences that we were talking about. And this is one skill that you need to know how to do, and that is identify an artery, vein and capillary on either a photomicrograph or an electron micrograph. And that's what we can see here. So a photomicrograph is an image taken using a light microscope or from a light microscope. An electron micrograph is an image from an electron microscope. So with the arteries, we can see an artery here and a vein there. The way that you can tell the difference is the artery wall is much, much thicker and the lumen is smaller compared to the veins. You will also typically see that the artery remains more circular, whereas the vein, the lumen can be distorted and you can actually squash the vein and you get this non-uniform or asymmetrical shaped lumen. The capillaries, which is what we're looking at here in the electron micrograph, have much, much thinner walls. And we can also see here, this is a red blood cell within the capillary, and it only just fits in, which we were talking about on the slide before. You have such narrow diameters of the lumen in capillaries so that red blood cells can only just fit through. In addition to being able to identify the blood vessels under the microscope, you also need to be able to identify the different components of the blood. So the different components of the blood that you need to be able to identify are red blood cells, monocytes, neutrophils and lymphocytes. And what we've got here is a diagram of each of those. And the red blood cells, which we can see down here on the bottom left, you can recognise these because they have that biconcave shape, and that dip in the middle is where we can see this darker image, or it's where it's dipping in, so it's almost like a shadow. Then we've got the photomicrograph of a lymphocyte. And to recognise these ones, with the lymphocyte, you don't have granular cytoplasm, so it looks slightly smoother, and there's a very large and pretty spherical nucleus. So that's how you can identify a lymphocyte. This one here is showing you a monocyte, and these are non-granular cytoplasm, but the nucleus looks a bit like a kidney bean, which we can see here, it's lobed and it's almost like a kidney bean shape. The one at the top then is a photomicrograph of a neutrophil, and this time it is granular cytoplasm, so you can see it looks a little bit dotty. And you also have a nucleus which is lobed and it looks more like a string of beads. So we've got multiple sections or lobes to that nucleus. So the tissue fluid then, we said this is one of the things that is occurring at the capillary beds, and this is where you have water and small molecules forced out of the capillaries, and they then surround the cells or the tissues. And it's all due to hydrostatic pressure and oncotic pressure. So hydrostatic pressure is the pressure exerted by a liquid. Oncotic pressure is the tendency of water to move into the blood via osmosis. And it's the interaction of these two that first of all results in the formation of tissue fluid and then results in the reabsorption. So let's have a look at the formation first of all. As the blood enters the capillaries, which we can see here, from the arterial end, there's a smaller diameter of the capillaries compared to the arterioles, but you have the same volume of liquid. And this results in a higher hydrostatic pressure forming. That high hydrostatic pressure forces water and small molecules such as glucose, amino acids, fatty acids, ions, and gases like oxygen out of the gaps between the cells that make up that endothelium layer of the capillaries. Now that liquid and small molecules that's forced out is then known as tissue fluid because it surrounds the tissues. And this is essential because those small molecules that we talked about, the glucose, amino acids, oxygen, those can now enter the cells by diffusion, or it could be active transport, and be used by the cell. Simultaneously, any waste products within the cell will be diffusing or moving out back to transport into the tissue fluid, and that waste can then be reabsorbed with the rest of the liquid from the tissue fluid back into the capillaries to be removed from the body. So at this point, the hydrostatic pressure is higher than the oncotic pressure, and we're talking about the arterial end here. And that's why the net movement of water is out of the capillaries to form the tissue fluid. When we get to the other end though, the venial end of your capillary, you will have reabsorption happening. And this is because, first of all, large molecules, so for example, plasma proteins, those don't get forced out of the gaps between the cells and the capillaries, and that's because they're too big to fit through. So they remain in the capillaries, and this lowers the water potential of the blood which remains in the capillaries.
So this lowered water potential will result in higher oncotic pressure towards the venial end of the capillaries. And simultaneously, you have a lower hydrostatic pressure because water has been forced out. So you have a lower volume of liquid within the capillary, and that's why the hydrostatic pressure is lower in comparison as well to the oncotic pressure. So that results in a net movement of the water moving back into the capillaries by osmosis. Now, once equilibrium of the water potential has been reached, so you have the same water potential in the capillaries and the tissue fluid, no more water can enter or re-enter the capillaries. And that final amount of tissue fluid then that can't be reabsorbed by the capillaries or into the capillaries is instead absorbed into the lymphatic system. And once it has been absorbed into the lymphatic system, it does eventually drain back into the bloodstream through a blood vessel near the heart. Once the liquid is in the lymphatic system, it's called lymph, and lymph has a very similar composition to plasma, except it doesn't contain those large plasma proteins or red blood cells, and it has less oxygen within it. Next, then we're going on to the transport of oxygen and carbon dioxide. So the transport of oxygen is done through the protein hemoglobin, and hemoglobins are groups of globular proteins found in different organisms, and each organism has a slight different protein. Hemoglobin has a quaternary structure, and hemoglobin is found in the red blood cells and it's responsible for transporting oxygen. So here's hemoglobin, and we can see it's made up of 4 polypeptide chains, and that's why it's a quaternary structure, because it contains more than one polypeptide chain. Myoglobin isn't hemoglobin, but it is a protein that also has a heme group and it can transport oxygen. But there's only one polypeptide chain, and it's in the muscle tissue invertebrates, and some organisms also have myoglobin. It has a very, very high affinity for oxygen, even at very low partial pressures. And we're going to come back to that concept. So oxyhemoglobin dissociation curves is the way that we can have a look at the affinity that hemoglobin has for oxygen. So on these graphs, we get the percentage saturation of hemoglobin with oxygen at different partial pressures. And you always get this S-shaped curve or sigmoid-shaped curve. And we can see that oxygen is loaded in regions with a high partial pressure and is unloaded in regions with a lower partial pressure. So this is your oxyhemoglobin dissociation curve. And the way we can actually tell that this loading and unloading happens is at these higher partial pressures, we can see that the hemoglobin is 100% saturated. So that means the hemoglobin must have a higher affinity for oxygen at high partial pressures of oxygen. Therefore, it loads the oxygen more readily and it's binding to the hemoglobin. And that's why it's fully saturated. At lower partial pressures, we can see that the saturation has dropped, so the oxygen must be unloaded from the hemoglobin. Now, this typically would be occurring in the respiring tissues, because in respiring tissues, oxygen is being used up for aerobic respiration, so there is a lower partial pressure of oxygen. Now, that's an advantage because if the affinity that hemoglobin has for oxygen is lower at respiring tissues, that means it will unload that oxygen and be available for aerobic respiration. The alveoli have high partial pressures of oxygen because that is where gas exchange is happening. So this is useful then that there's a higher affinity, because that means the hemoglobin will then be able to load up with all of that oxygen and become fully saturated to transport it to the respiring tissues. These graphs also demonstrate cooperative binding. And this cooperative nature of oxygen binding to hemoglobin is due to the hemoglobin changing shape when the first oxygen binds. So the reason there is a lower affinity at these lower partial pressures is the binding sites aren't as exposed when you don't have any oxygen bound to it. But as soon as the first oxygen binds to hemoglobin, it slightly changes the shape of the protein to reveal those binding sites. And that then makes it more likely for the subsequent oxygens to be able to collide and bind with hemoglobin. And that's what explains that sigmoid shape graph. Carbon dioxide also has an effect on hemoglobin's affinity for oxygen, and this is known as the Bohr effect. The Bohr effect is when a high carbon dioxide concentration causes the oxyhemoglobin curve to shift to the right. And what this means is, even at the same partial pressure, so for example at 20, when the curve is shifted to the right, you have a lower saturation than you would if the curve was shifted to the left. So even at the same partial pressure of oxygen, you have a lower affinity, which means that it's going to be unloading the oxygen more readily. The advantage of this is, if you have a high concentration or partial pressure of carbon dioxide, the fact that hemoglobin has a lower affinity for oxygen is good, because that would indicate that there must be respiration occurring. Because carbon dioxide is made in respiration, that's an acidic gas, and it's changing the shape of the hemoglobin so it unloads the oxygen more readily. So that means oxygen is being delivered to the site of respiring tissues so that aerobic respiration can continue for longer and more ATP will be created. There are also differences between the affinity of hemoglobin depending on the organism itself and how it's adapted to its environment. And that's what we're going to have a little look through. So a human fetus, we're going to have a little look at here. So here we have the fetal hemoglobin compared to adult hemoglobin, and we can see it's shifted to the left. Now, if a curve shifts to the left, that indicates a higher affinity for oxygen. And the way we can tell that is, even at the same partial pressure, so let's say 25, we can see that when the curve shifts to the left, there is a higher saturation of the hemoglobin with oxygen. And that's an advantage because the fetus gets its oxygen from the mother's hemoglobin as it transports around in the blood, in the red blood cells, through the placenta. So it needs to have, the fetal hemoglobin needs to have a higher affinity than the hemoglobin of the adult so that it can essentially take that oxygen from the adult hemoglobin. Another example is animals that live at high altitude. So for example, a llama lives at a high altitude where there are lower partial pressures in that particular environment. So there's lower partial pressures of oxygen. So for a llama, they also have hemoglobin with a higher affinity, so that even at very low partial pressures, which is what their environment is like, their hemoglobin can still become loaded, saturated, and they're able to get that oxygen for aerobic respiration. Another example is a dove or any other smaller organism which has a high metabolic rate. Because they have a faster metabolism, they require more oxygen for continual aerobic respiration, or faster rates of continual aerobic respiration. And that is so that they can release the energy or create ATP for contracting muscles. So they actually have a curve that shifts to the right, indicating a lower affinity. The hemoglobin has a lower affinity for oxygen because even at the same partial pressure, we can see it's got a lower saturation, and that means it's going to be unloading oxygen more readily, which means the oxygen's being delivered and unloaded to respiring tissues so that aerobic respiration can continue. Earthworms and other organisms that might burrow and live underground, or it could be deep diving animals, they are at lower partial pressures of oxygen in their environment. So they need to have a higher affinity, so the curve is shifted to the left, so that even in those low partial pressure environments, the hemoglobin can still become saturated with oxygen. Next then we have a look at the transport of carbon dioxide. And there are 4 ways that carbon dioxide can be transported. One is dissolved in the blood plasma. Next we have as hemoglobinic acid, and carbon dioxide can react reversibly with amino acids in hemoglobin to form hemoglobinic acid. It can also be in the cytoplasm of red blood cells in the form of hydrogen carbonate ions. And also it could be the formation of carboaminohemoglobin. Now, almost 85% of the carbon dioxide is transported as hydrogen carbonate ions in the red blood cells, which is what we can see here with this reaction. Water and carbon dioxide react in a reversible reaction, and that forms hydrogen ions and hydrogen carbonate ions. Carbonic anhydrase is an enzyme in the cytoplasm of the red blood cells, and that catalyzes that reaction. The carbonic acid can move out of the red blood cells by diffusion, and in exchange, chloride ions diffuse into the red blood cells. Both of these ions are negative, so these exchanges maintain the electrical balance of the red blood cells, and this is known as the chloride shift. Carboaminohemoglobin is formed when carbon dioxide binds to the amino groups of globin proteins in the hemoglobin. And this reversible process occurs in tissues where carbon dioxide levels are higher, facilitating the transport of carbon dioxide from tissues to the lungs. The binding of carbon dioxide to hemoglobin helps to remove carbon dioxide from tissues and transports it back to the lungs for exhalation and therefore to be removed by gas exchange from the body. Finally then, we're going to have a look at the heart. In particular, we're looking at the mammalian heart. So the heart is an organ. It's made up of cardiac muscle, which is a specific type of muscle only found in the heart. And it's myogenic, which means it automatically contracts and relaxes. It doesn't require an input from the nervous system to make it contract and relax. It does require the nervous system to control the heart rate, but we're going to come to that later in this video. It also never fatigues, unlike skeletal muscle, which can fatigue. Cardiac muscle, as long as it has a constant supply of oxygen and glucose, it will always contract and relax. And that's the role of the coronary arteries. They supply the cardiac muscle with oxygenated blood and glucose so that they can respire aerobically, produce ATP, and therefore the muscle can continually contract and relax. The heart is also surrounded by a pericardial membrane, and these are inelastic membranes which prevent the heart from filling and swelling with blood. So then if we have a look at the inside of the heart, there are 4 chambers. You've got the 2 ventricles, the left and the right, and then the 2 atria, the left and the right. And you might notice that literally this would be your right side if we're looking at the picture, but we've labelled it as the left. And that's because you've always got to imagine it as if it was on you. So if you were to pick up that picture and hold it against your chest, the bit coloured in red would actually be on your left side. So that's how we label the heart, as if you were holding it up against your chest, and that would be on the left. And the left ventricle, which we can see here, has a much thicker cardiac muscular wall, and that means it can contract with more force and pump the blood out of the heart at higher pressure. And the blood leaves the left ventricle through the aorta to go to the rest of the body. So that's an advantage because we need the blood to be forced out at higher pressure so that it will then be able to reach all of the respiring tissues in the body. The right ventricle is only pumping blood out of the heart to the lungs, which is much closer. And also you don't want a really high blood pressure in the lungs because it could cause damage to the capillaries. So for that reason, the cardiac muscle in the walls of the right ventricle are much thinner because they don't need to contract with as much force and the blood is not pumped out at as high a pressure. Both of the atria, the right and the left, have much thinner muscular walls, and that's because they don't need to contract with much force at all because they're only pumping blood from the atria down with gravity into the ventricle. Next time we're going to have a look at the cardiac cycle, and this links together the contraction of these different muscles in the walls and also the valves, the atrioventricular valves, which are between the atria and the ventricles, and the semilunar valves, which are between the 2 ventricles and the arteries carrying blood away from the heart. And it's split into 3 key stages. We have diastole or diastole, people pronounce this differently, atrial systole or systole, and ventricular systole or systole. So as I said, everyone pronounces this slightly differently depending on where you're from. I tend to say both just to be user-friendly for anyone that happens to be listening. So here we have our cardiac cycle. And first of all, we can see both the atria and the ventricles are in diastole, which means the cardiac muscle is relaxed. That's what diastole or diastole means. That is when the muscles in the heart are relaxed. And when they are relaxed, that means that all of the chambers are slightly larger in volume. Therefore, the pressure inside of them is lower. And that is what causes the blood to enter the two atria at this point. As the blood starts to enter the atria, that increases the pressure in those atria. And it will get to the point where it increases them to a point where the pressure in the atria is higher than the pressure in those empty, relaxed ventricles. And that forces open those atrioventricular valves. We then get to the point where we have atrial systole, and this is when the muscles in the atrial walls will start to contract, which increases the pressure further and will force the blood from the atria into the ventricles. Then the atria will relax and we go into ventricular systole, which is when the muscles in the walls of the ventricles contract. And at this point, because the muscles are contracting, that will then result in a higher pressure in the ventricles compared to the atria. And that causes the atrioventricular valves to shut. And when the muscles are contracting even more, it increases the pressure further, so that the pressure is higher in the ventricles compared to those two arteries, the pulmonary artery and the aorta, forcing open the semilunar valves. So the blood now carries on flowing in one direction from the ventricles up and out of those two arteries. Can't go back into the atria because those atrioventricular valves are shut. And that is the role of these valves. It ensures that the blood flows in one direction through the heart. And that's all controlled by the pressure changes between the different chambers and the arteries. So here then we've got that information that I was just talking you through. So if you do want to pause, make notes or flashcards, you can do that. Diastole or diastole, the atrioventricular muscles are relaxed. This is when blood will enter the atria via the vena cava and pulmonary vein. And the blood flowing into the atria increases the pressure with the atria. Atrial systole, that is when the atriomuscular walls contract, increasing the pressure further, which causes the atrioventricular valves to open and blood will then flow into the ventricles. But at that point, the ventricles are relaxed or in diastole. And then lastly, we get that ventricular systole, when after a short delay, the ventricular muscle walls then contract. That increases the pressure beyond that of the atria, causing the atrioventricular valves to close and the semilunar valves open. And the blood is pushed from the ventricles into the arteries, which are either the pulmonary artery or the aorta. Now, you could be asked to do this particular math skill, which is calculating the cardiac output. And that is your heart rate times the stroke volume. And the heart rate is the number of times your heart beats per minute. And the stroke volume is the volume of blood that leaves the heart after each heartbeat. So essentially your cardiac output is the volume of blood which leaves one ventricle in 1 minute. And that is how you'd go about calculating it. Or you could be asked to rearrange the equation to work out heart rate or stroke volume as well. So what is controlling the speed at which the heart is contracting and relaxing and therefore pumping the blood? And that's what we're going to go on to. So we'd already said that cardiac muscle is myogenic, so it doesn't require input from the nervous system to make it contract and relax, but the rate at which it contracts is controlled by electrical activity and the nervous system. And the key structures are the sinoatrial node, which is located in the right atrium, and it's also known as the pacemaker. You then have an atrioventricular node, the AVN, which is located near the border of the right and left ventricle, but it's still within the atria. Now, within this picture, it's here, but really it's quite a bit closer to the septum. The bundle of His runs down the septum and up the walls of the ventricle, and then the Purkinje fibres are also going up the walls all the way around, so it's going to reach all of that cardiac muscle. And all of these structures can conduct electrical activity. So the SAN releases the wave of depolarization, which is the electrical activity. The AVN can then transport that wave further, and then it transports down the bundle of His and the Purkinje fibres. And when this electrical activity or depolarization touches cardiac muscle, it causes it to contract. So let's have a look at this process. The first thing that happens is the SAN, the pacemaker, releases a wave of depolarization across the two atria, and that causes the atria to relax. So that is our atrial systole. Now you do have, across the division between the atria and the ventricles, a layer of non-conductive tissue. So that means that that wave of depolarization can't go directly down into the ventricles, and instead when it hits the AVN then releases another wave of depolarization. And that causes a slight delay between when the atria contract and when the ventricles will then be able to contract. And that's an advantage because it means that there is a time delay between when the blood is pumped from the atria and then when the ventricles contract. So essentially there's time for the ventricles to fill up with all that blood before they contract. And that's what we can see here. Instead of that wave of depolarization going directly down into the ventricles, it will be picked up by the AVN. The AVN releases the wave, which then goes down the bundle of His, up the Purkinje fibers, and then it will cause that ventricular muscle to contract. And the contraction happens at the apex of the heart first, which is the very bottom. And that then means it will contract from the bottom all the way up the walls of the ventricles, and it should force out all of the blood from the ventricles. Then we get to this diastole stage, and that is when repolarization happens, and that is when you don't have that electrical activity anymore causing the muscles to contract. So instead, all of the cardiac muscle relaxes. CIE topic 9, and in this one we're going through gas exchange. So the mammalian gas exchange system, then, you need to know the key structures, the trachea or trachea, bronchi, bronchioles, and the alveoli. And the route that air takes during ventilation is, it will be inhaled through the nasal cavity or mouth, then it passes down the trachea, which is the windpipe. It then is split between these two tubes, which are the bronchi. It then branches into multiple smaller tubes, which are the bronchioles, and then we get to these air sacs at the end, which are the alveoli. So let's have a look at these structures in more detail. Now, you could be asked to identify these structures on a photomicrograph or an electron micrograph. So we're going to go through these structures, what their function is, but first I'm just going to talk you through what to look for to be able to identify them. So for the trachea, you're looking for a large tubular structure which is lined with ciliated epithelium, which you might be able to see on an electron micrograph, so those hair-like structures coming out of the cells. You may also see goblet cells, which are interspersed amongst epithelial cells, which would be this big, almost like indent into the cell where you have mucus being produced. The tracheal wall should contain cartilage as well, and you get these C-shaped rings of cartilage. The bronchi are similar to the trachea, but smaller diameter. So the bronchi are also lined with ciliated epithelium. They contain these cartilaginous plates instead of complete rings, and the plates may appear as irregular shaped segments along the bronchiole wall. The bronchioles are smaller, less structured airways compared to the trachea and bronchi. The bronchioles are lined with simple ciliated epithelium without the presence of cartilage. You have smooth muscle fibres which may be visible in the bronchiolar wall, appearing as irregular bands or layers. Now the alveoli, there's lots of round structures with an air space in the middle, and they've got very, very thin walls. They're only 1 cell thick. So if we go through these structures in more detail than just what they would look like on a microscope. So we're starting with the trachea, which is the air pipe or windpipe, and you have these C-shaped rings of cartilage, which is what you can see here in this beige colour. And that cartilage provides structural support so that the airway is constantly open and it doesn't get flattened or stuck together. And it's C-shaped rather than the whole way round because your esophagus would be here, and you don't want that hard cartilage against the esophagus because as the esophagus is contracting and relaxing with peristalsis, that could be interfered by that cartilage. You also have ciliated epithelium with goblet cells. The cilia, those hair-like structures, are there to sweep mucus up and out of the lungs to prevent infection of the lungs. And then it's the goblet cells that produce that mucus. So goblet cells' upper section swells with mucin droplets. Mucus comprises mucin, and it's this viscous solution of glycoproteins with multiple carbohydrate chains, making it really sticky and capable of trapping inhaled particles, which could be dust or it could be pathogens. Mucus glands beneath the epithelium produce the mucus. Certain chemical pollutants like sulfur dioxide and nitrogen dioxide can dissolve a mucus, creating an acidic solution that irritates the airway lining. So that's not a good thing. Smooth muscles is within the walls of the trachea. So smooth muscles are within the walls of the trachea. The muscle contracts if there are harmful substances detected in the airway. And this results in the lumen of the trachea constricting and reducing airflow to the lungs as a way to try and protect the lungs. When the smooth muscle relaxes, the lumen dilates. This stretch and recoil of the lumen is possible due to the elastic fibres within the tracheal wall. Next, in the bronchi and the bronchioles, the trachea splits into two tubes, the bronchi, so you have a left and a right one, and that leads to the left lung and the right lung. And those bronchi split into the bronchioles, which are these smaller tubes. And both the bronchi and the bronchioles have cartilage within their walls for structural support to keep the tubes open. Now, you will have noticed in that previous slide we said that the bronchioles don't have cartilage. They will have cartilage at the very top parts, but as they branch further and further, those smaller tubules in the bronchioles don't have cartilage. Then the bronchioles end at the alveoli, which are these air sacs, and they're located at the ends. It's the site of gas exchange. Oxygen diffuses from the alveoli, where you have a high concentration of oxygen or partial pressure, into the blood, where there is this lower partial pressure of oxygen. And carbon dioxide does the opposite, 'cause it's also moving down its concentration gradient. The blood that is entering the capillaries at this point is a high concentration of carbon dioxide, so it diffuses from the blood into the alveoli where it will be exhaled. So in terms of adaptations then, first of all, the fact that there are so many alveoli in both lungs is what provides a large surface area. One individual alveoli doesn't produce a large surface area. It's the fact that there are millions in both lungs. The short diffusion distance, though, that is created by the fact that the alveoli wall is made up of just one thin layer of cells, and those thin layer are called squamous epithelial cells, which I love that word. Squamous is such a cool word. Basically, it's like the idea it's squashed. It's a squashed, flat cell, and that reduces the diffusion distance. Also, the endothelium of the capillary is just one layer of cells as well, so you've got a very short diffusion distance. The gases only have to diffuse through two cells, which are both flat. The maintaining of the concentration gradient, that's done through ventilation, but also the fact that the blood is constantly being transported away. So as soon as the oxygen diffuses into the blood, it's being transported away. So a bit more then about this gas exchange in the alveoli. We've said that oxygen is inhaled into the alveoli and diffuses across the layers of those two structures. But we haven't said yet that the fact that the moist lining of the alveoli is there. So you have this moist layer on that layer of cells, and that's so the gases can dissolve in that liquid and then they diffuse across that way. We did talk about that single layer of cells, which is squamous epithelial cells. They're flat, they've got that really short diffusion distance. The oxygen then diffuses through the endothelial cells lining the capillaries. Simultaneously, carbon dioxide, which is the waste product of respiration or aerobic respiration, diffuses from the blood in the pulmonary capillaries into the alveoli, and carbon dioxide diffuses in the opposite direction of the oxygen moving in. The efficiency of gas exchange is due to those three factors that we talked about. The large surface area, because there's so many alveoli, the short diffusion distance because of these squamous epithelial cells, and the fact that both the alveoli wall and the capillary endothelium is only made up of one single layer of cells, and the concentration gradient is maintained by ventilation and that blood flow in the capillaries. Next, then, you need to know about the distribution of tissue and cells in the gas exchange system. We've talked about this throughout, but we've got a summary here for you, so you can just create a summary sheet of information. So the cartilage is distributed in the walls of the trachea and those larger bronchi, providing structural support so that it doesn't collapse, those tubes don't flatten and collapse. The ciliated epithelium, that's in your nasal cavity, trachea and bronchi, and it's to move the mucus, to sweep that mucus up and out of the lungs. Goblet cells, you have lots of them in the respiratory epithelium, secreting mucus to trap and remove inhaled particles. The walls of the alveoli, we said, consist of a single layer of squamous epithelial cells, and that allows for the efficient gas exchange between the alveoli and the capillaries. Smooth muscle is found in the bronchioles, and that helps to regulate airflow by controlling the diameter of the airways. And the capillaries form a dense network surrounding the alveoli in the lungs, which helps to facilitate rapid gas exchange because it maintains that concentration gradient.
Topic 10, we're going to be looking at infectious diseases first and then antibiotics. So within the infectious diseases topic, you need to know that bacteria, viruses, protoctista and fungi are able to cause these infectious diseases. But you actually focus on these three in particular for your exam board. So pathogens can cause harm through directly damaging tissue or through the release of toxins. And the three particular diseases you need to know caused by these pathogens are tuberculosis and cholera for bacteria, HIV, which can develop into AIDS, and then malaria as a protoctista disease. So we're going to go through each of these, starting with the bacterial diseases and tuberculosis. So this is caused by two different strains, Mycobacterium tuberculosis and Mycobacterium bovis. It can infect humans, deer, cows, pigs, badgers, and it causes harm, the bacteria causes harm, by damaging the lung tissue and suppressing the immune system. It can be cured though using antibiotics because it's caused by bacteria, and it can be prevented in the first place if you have a vaccine, so you develop artificial immunity to it. So let's have a look at it in a bit more detail. Tuberculosis spreads mainly through the air when infected individuals cough, sneeze or speak, releasing the Mycobacterium tuberculosis bacteria in tiny droplets. Transmission usually requires close and prolonged contact, particularly in crowded or poorly ventilated settings. And factors like having a weakened immune system increases your risk of developing the symptoms, and therefore you're able to transmit the disease as well. So TB, tuberculosis, can also remain latent in the body without causing symptoms. Next then, the Mycobacterium bovis, which is the other strain. This is the one that is found in animals such as cats and badgers, and it poses a risk to humans through direct contact or consumption of contaminated food. Now, control measures include testing, vaccination, and other biosecurity practices, such as trying to prevent the spread through having more space. And this is crucial to prevent that spread among animals and reduce human infection. So a bit more on the prevention and control. So biological factors then, the mycotuberculosis bacteria is primarily transmitted, as we said, through air droplets. And understanding that TB transmission can help then to reduce the risk factors and infection, because you can do simple things such as coughing into your hands, washing your hands. You could also do quarantining people who are infected. If we have a look at social factors, social economic factors such as poverty, overcrowding, malnutrition, because that lowers or weakens your immune system, and limited access to healthcare, so maybe you aren't vaccinated, could contribute to the transmission. So TB control efforts should address social factors as well, so trying to alleviate poverty, maybe providing vaccines for free, improving living conditions, and that should help prevent and control the transmission of TB. Next, on the economic factors, you need to have adequate funding of TB control programs. So, for example, diagnosis, treatment with the antibiotics, and preventing with the vaccines are all essential to try and reduce the incidence of people getting the disease and dying from it. So strengthening healthcare systems, including laboratory infrastructure, the ability to diagnose, all of these will be crucial to help prevent and control TB. The next bacterial disease is cholera, and this is caused by Vibrio cholerae, and it's a highly contagious pathogen that thrives in contaminated water and food sources. It spreads primarily through the consumption of contaminated water or food, particularly in areas where there's poor sanitation and hygiene. It infects the small intestines, where it produces toxins that leads to severe diarrhea and vomiting. And without treatment, cholera can rapidly lead to dehydration and even death, making it a significant public health concern, especially in regions lacking access to clean water and proper sanitation facilities. So there are efforts to prevent cholera outbreaks, and this often focuses on improving sanitation, promoting hygiene practices, and providing access to safe drinking water. And this slide here just goes through a bit more detail on those, broken down into biological, social, and economic factors. So one way that's helped to prevent and control the transmission was understanding the biology of it, because now we know that it's transmitted through contaminated food and water and that it's a bacteria. We know that you have to make sure you're drinking water that has been completely cleaned and food is thoroughly cooked, and you can treat it with antibiotics. Social factors as well, knowing that drinking or having access to clean water is essential. Improving hygiene practices such as washing hands can also have a huge role in trying to prevent these spreads, as well as education and having communities aware of how it is transmitted. Lastly, then we've got the economic factors. So again, it's linked into the idea of poverty, poor infrastructure. If you can improve all of those, it's going to help reduce the spread and contraction of cholera. Next, then we have a look at viruses, and viruses are non-living, they're acellular, which we talked about in earlier topics and videos. They're smaller than bacteria, and they just consist of genetic material, which could be DNA or RNA, a capsid, and an attachment protein. The viral replication happens inside of host cells, and the virus has to inject its nucleic acid into the cell of the host to be able to replicate. And bacteriophage are actually an example of viruses that infect bacteria. Now, the key example that you need to know is the virus HIV, and it consists of the following structural features: a capsid, which we can see here as these the dark blue circles, and that is an outer protein coat surrounding the genetic material. That is what is in the core, which is the genetic material, which is RNA for HIV. And the enzyme reverse transcriptase is also within that, which is needed for the viral replication. There is an envelope layer, which is an extra outer layer made out of lipids, which have been taken from the host's cell membrane. And then we have the protein attachments, and these are on the exterior of the envelope to enable the virus to attach to the host's helper T cells. So if we have a look then at how HIV is transported and how infection occurs, it's transported in the blood, and it attaches to CD4 proteins, which are these receptors on the outside of helper T cells, which are part of the immune response. The HIV protein capsule can then fuse with the helper T cell membrane, and that enables the RNA and the reverse transcriptase that were within that capsid to enter, and it's entering the host cell. The HIV enzyme reverse transcriptase, which has been entered along with that RNA into the host cell, are then copied into DNA, and that DNA copy can move into the nucleus of the helper T cell. And once it is in the nucleus of the helper T cell, that host's DNA as well as the viral DNA will be transcribed, translated, and so the viral proteins are going to be made along with the other proteins that would typically be made in the helper T cell. And in that way, it can then reassemble all of those proteins and a new viral particle has been created. So someone is described as HIV positive when they're infected with HIV, but it's only described as AIDS when the replicating HIV viruses in the helper T cells have interfered with the normal function of the immune system. So if I just go into a bit more detail on this, the point above here where we talked about how HIV replicates inside of the host cell, once all of those viral proteins have been assimilated to make a new virus particle, when you have enough new virus particles in that helper T cell, the helper T cell gets destroyed. And if you have lots of helper T cells being destroyed, that then means that your immune system is compromised and it won't function normally. And that is when someone has AIDS. So with the helper T cells being destroyed, that means you are unable to have this immune response and defend yourself against other infections and even cancer. And that's why HIV, when it's destroyed enough helper T cells to result in AIDS, can cause death. So the HIV itself doesn't cause death, but when you've developed AIDS, because the helper T cells have been destroyed, you can then die from other infections because you can't defend yourself. So here then we have prevention and control methods for HIV. So first of all, it was understanding HIV. So we know now that it's transmitted through sexual contact, blood-to-blood contact, so for example, sharing needles, and perinatal transmission. So that means through the placenta from mother to child. And understanding this means that we can then put in place preventative methods to try and stop that transmission. So, for example, having barrier methods as contraception, so condoms, for example, blood-to-blood contact and not sharing dirty needles as well. Social factors then, we've got stigma, discrimination, social marginalisation of key populations, including, it has been in the past, sex workers, men who have sex with men, people who inject drugs, has happened. But it's best to actually think about these biological factors of how you can protect people without having this discrimination. Economic factors as well. So again, poverty always plays a role that links to the lack of education that people might have of how they can protect themselves, but also it might link to lack of access to drugs, so antiretroviral therapies or ART, which an infected mother can take while they're pregnant to prevent it being transmitted to their child. Then we look at the protoctisters. So the protoctista or protist are eukaryotes. They're single-celled organisms or cells grouped into colonies. Very few are actually pathogenic, but the few that are incredibly dangerous. So the pathogen protoctisters are parasites and are usually transmitted through a vector. So we're going to be looking at malaria, which is transmitted by mosquitoes. So this is the example you need to know, malaria. And it's caused by the protoctista Plasmodium, and it's spread to humans through the vectors mosquitoes. Plasmodium reproduce both sexually and asexually within mosquitoes and within the human host. So it's passed from mosquitoes to humans when mosquitoes bite and take blood from humans. In humans, the Plasmodium infects the red blood cells, the liver, and the brain. There are some preventative medicines, but no vaccine and no cure. And we can see here a range of the different symptoms that this Plasmodium causes once it's infected a human. So how it can be prevented and controlled? The main way it's prevented is through preventing the vectors, which are the mosquitoes. So having that understanding of the life cycle enabled us to realise that the transmission through the vectors could be a key way to prevent humans getting infected. So this again links to social factors, economic factors. To prevent that transmission, you can have medicines in advance, like antimalarial drugs, to try and prevent you getting the disease. But having things like insecticide treatments to kill those vectors, the mosquitoes, or having bug spray to try and prevent yourself being bitten, having mosquito nets to prevent them being able to get to you when you're asleep at night in countries that have lots of mosquitoes as well. Next, we go on to antibiotics. So an antibiotic is a substance produced by a microorganism and it inhibits the growth of other microorganisms. Since the discovery of penicillin, which was the first antibiotic discovered in the mid-20th century, antibiotics have been used widely to treat bacterial infections and save many lives. So the way that antibiotics kill bacteria, because they do only kill bacteria, are by preventing cell wall synthesis. And this could be that it inhibits the enzymes responsible for making molecules in the cell walls, and the bacteria then die from the leakage of the contents or lyse from too much water entering. Or it could be through disrupting cell membranes. So it could be that it binds to the phospholipids in the bacterial cell membrane to distort the structure, and it makes the membrane too permeable so it bursts and therefore the bacteria would die. And the final way is they can interfere with protein synthesis. So it can attach onto bacterial ribosomes, which prevents the protein synthesis, and therefore if the bacteria doesn't have the proteins it requires, it will end up dying. So antibiotics only work against bacteria and not viruses, because viruses don't have cell walls. So those mechanisms that we just described of how that antibiotic can kill bacteria wouldn't work. And unlike bacteria, viruses lack cellular machinery for metabolism, growth, or reproduction. And again, that's why it doesn't affect them. Since viruses lack those metabolic pathways, that is why the antibiotics won't cause the destruction of them, as well as the fact that viruses are within the host cell themselves. So you'd have to destroy the host cell as well as the virus to be able to treat them. Now, antibiotics is not a perfect medicine because antibiotic resistance has developed in bacteria. And this has happened through random mutations that happen in the DNA of bacteria. And by chance, a mutation would have occurred, which results in the production of a new protein that provides a selective advantage, meaning it creates some protein, which now means that antibiotic cannot kill that particular bacteria. And as a result, that bacteria will survive, reproduce, pass on that mutated version of the gene, which creates a protein that enables them to survive, until eventually you have this widespread population that all have the antibiotic resistant gene. Now, this was really sped up by the widespread use and misuse of antibiotics, because the antibiotic was the selection pressure in this example of natural selection. And the more you were taking the antibiotics, the more that meant the non-resistant bacteria died, the resistant ones survived, and then they had no competition. So then they could really thrive. So they weren't competing for resources, water, and that's why the non-resistant ones were able to thrive inside of the host. And as a result, you had even more reproduction and passing on of that resistant gene. So that resistant bacteria reproduces rapidly until you have an entire strain of bacteria that are now resistant to that antibiotic. Now, the two most common resistant bacterial strains are Clostridium difficile and MRSA. Now, MRSA is actually a strain of bacteria resistant to multiple antibiotics, because once this process happened once, another random mutation happened in one of those bacteria, and this whole process happened again and again and again until you had this strain of bacteria resistant to multiple antibiotics. So the consequences of antibiotic resistance and prevention then. Antibiotic resistance poses a significant consequence for public health and healthcare systems and economies worldwide. Resistant infections are harder to treat because you don't have the antibiotics to kill the bacteria. And that can lead to increased illness, deaths, and healthcare costs. Limited treatment options exacerbate the problem, as healthcare providers may resort to broad spectrum antibiotic use, and that further increases the resistance because you're now adding in even more or adding another selection pressure. Moreover, the antibiotic resistance also undermines infection control efforts, increases the risk of outbreaks, and compromises the effectiveness of medical procedures like surgery and chemotherapy. So to reduce the impact of antibiotic resistance, you have different things that are occurring. So for example, doctors are now less inclined to prescribe antibiotics unless you really, really need them. So maybe you have a weakened immune system, so your immune system won't be able to fight the pathogen by itself. There's lots of awareness campaigns as well to educate people on the importance of not taking antibiotics unless they've been prescribed, not taking them for viral infections because they'll have no effect, and making sure you take the entire course of antibiotics that you have been prescribed to take as well. So let's have a look at topic 11. And topic 11 covers the immune system, antibodies, and vaccination. And we're starting with the immune system, looking at phagocytosis. Now, phagocytes, so for example, the macrophages and neutrophils, which are two types of blood cells, travel in the blood and squeeze out of capillaries to engulf and digest pathogens. And that's what phagocytosis is. It's a non-specific response whereby any foreign material that's detected will get destroyed by the phagocytes. And here we see the process of phagocytosis. Damaged cells and pathogens release cell signaling chemicals, which are the cytokines, and they attract the phagocytes to the site of infection. Opsonin protein can attach to pathogens to mark them and make it easier for neutrophils and macrophages to then engulf them. Phagocytes have receptors which can attach to chemicals on the surface of the pathogen, and the pathogen then surrounds and engulfs the pathogen into vesicles to create a phagosome. Within the phagocytes, there are lysosomes which contain hydrolytic enzymes, and those lysosomes will then fuse with the phagosome to make a phagolysosome, and it exposes the pathogen to that enzyme, the lysozyme, which hydrolyzes the pathogen, and any soluble useful molecules are absorbed into the cytoplasm of the phagocyte. The phagocytes will present the antigen of the digested pathogen on their surface, and they then become antigen-presenting cells. The second line of defence involves lymphocytes, and this time it is specific, and the lymphocytes respond specifically to particular shaped antigens. There's two types of lymphocytes, the B and the T lymphocytes. Both are created in the bone marrow, but the B cells mature in the bone marrow, which is why they're called B lymphocytes, whereas the T cells mature in the thymus, which is why they're called T lymphocytes. So we're going to have a look at the cell-mediated response, which is the response which involves T cells. It's called the cell-mediated response because it involves these T lymphocytes binding to antigen-presenting cells. So it's binding to cells themselves, which happen to have the antigen on their surface. And when you have the receptors on the T cells binding to antigen-presenting cells, it causes the T cells to divide rapidly by mitosis, which is this clonal expansion, because mitosis results in identical copies of cells. So you get this large number of T cells being created, which are all identical, and they're identical in that they contain exactly the same receptor on their surface, complementary to this particular antigen. Antigen-presenting cells are cells that present a non-self antigen on their surface, and that could be infected body cells presenting viral antigens on their surface, a macrophage which has engulfed and destroyed a pathogen presenting the antigens on their surface. Cells of a transplanted organ will have different shaped antigens on their surface compared to your self-cell antigens. And lastly, cancer cells will also act as antigen-presenting cells because they have abnormal shaped self-antigens on their surface. So let's have a look then at this whole cell-mediated response. Once a pathogen has been engulfed and destroyed by a phagocyte, the antigens are positioned on the cell surface membrane, which we saw, and we now call an antigen-presenting cell. T helper cells have receptors on their surface, and those can attach to complementary shaped antigens on the surface of these antigen-presenting cells. Once those attach, interleukins are produced, which activates the T helper cells to start to divide by mitosis, and that's when they'll then replicate and make large numbers of clones. So lots of these T helper cells with that particular shaped receptor. Those clone T helper cells then differentiate into different types of T cells. You'll have some which will differentiate into even more T helper cells to produce more interleukins to keep this process going, and also to activate B lymphocytes. Some produce interleukins to stimulate macrophages to perform more phagocytosis. You also get T memory cells, which will store a memory of that particular shaped antigen. And then T killer cells, or cytotoxic T cells. And those ones we'll have a look at in a bit more detail. T regulator cells will suppress the immune response to ensure the cell-mediated response will stop, and therefore it's only occurring when the pathogen is present. Because you don't want this response happening all the time. You only want it to happen when you are infected with a particular pathogen. So the C killer cells, or those cytotoxic T cells, these are the T cells that destroy abnormal or infected body cells. And they do this by releasing a protein called perforin, which embeds into the cell surface membrane, and it makes a hole, a pore, so that any substances can enter or leave the cell, and that is what causes cell death. Because either you have too much liquid and substances leaving the cell, causing it to dehydrate and die, or you have too much liquid entering, and that causes it to burst. So this is most common in viral infections because the virus infects the body cells, and by destroying the body cell and sacrificing your body cells, it prevents the virus being able to replicate further. Next, let's have a look at the B cells, and those are part of the humoral response. So B cells are activated by T helper cells, and this is done by the T helper cells binding to B cells with complementary antibodies to antigens. And this is clonal selection, because the T cells will only bind to B cells which do have a complementary shaped antibody on their surface. So it is selecting the correct shape, that complementary shaped B cell, and then they get cloned in mitosis. And that's what we're going to have a look at here. So that B cell is activated by the release of interleukins from that T helper cell once they're bound. That causes the B cells to divide by mitosis to make large numbers of clones which have that exact shape antibody on their surface. So that is your clonal expansion. The B cells then differentiate into either memory B cells or plasma cells. The plasma cells are the ones that can produce antibodies, and the antibodies attach to the antigens on the pathogen to help destroy them by agglutination, and that's coming up later in this video. They also mark them so that phagocytes can locate them and destroy them. This is a primary immune response, which means it's the first time that you are encountering that pathogen. And you store these memory B cells, which remain in the blood for a long time after infection. So if you are then reinfected with the same pathogen with that same shaped antigen, the B cells can rapidly differentiate into plasma cells and produce large numbers of antibodies rapidly, so that you can then destroy the pathogen before it causes any symptoms. So that's your primary and secondary immune response. The primary response is the first time you're ever exposed to that pathogen and antigen. And because it's the first time, it can take a few days for the lymphocytes to create enough of the correct complementary shaped antibodies to help to destroy the pathogen. And therefore, the time that takes, the pathogen is dividing, causing damage, and therefore you have symptoms of the disease before the pathogen is destroyed. In a secondary immune response, because you're being reinfected with the same pathogen, the memory B cells can produce large amounts of antibodies rapidly so that the pathogen is destroyed before it can cause any symptoms. And that is what active immunity is. You're immune because you've been actively infected with this pathogen. Next, then, we have a look at antibodies and vaccination, carrying on from this idea of immunity. So antibodies are proteins, and it's those T helper cells that we saw stimulate the B cells to produce interleukins, initiating the humoral response, which results in the production of plasma cells, which produce antibodies. So antibodies are globular proteins. They're quaternary structures, and we actually have 4 polypeptide chains. We have 2 lighter chains, which are shorter, and 2 heavy chains, which are longer. The binding sites are up here, and that is where you have this unique shape, which will be complementary to the shape of a particular antigen. And that's where a particular antigen can bind to a particular antibody. The rest of the antibody is constant, so it'll be exactly the same shape in all antibodies. And when an antigen binds to an antibody, we call that an antigen-antibody complex. And this is where it links to agglutination. So antibodies can work in 3 ways: agglutination, or through marking pathogens, and also acting as antitoxins. Agglutination is what we can see in this image. It's the clumping together of pathogens to make it easier for phagocytes to locate and then engulf the pathogen all in one go. Antibodies also act as an opsonin when an antibody-antigen complex has been formed. The antibodies are marking the antigen, making them more susceptible and more easy to locate for phagocytosis. And then also antibodies can bind to toxins, preventing them from entering the cells and causing harm. And in this way, they're acting as antitoxins. Now, monoclonal antibodies is a technology that has been used based on your knowledge or our knowledge in science of antibodies. And the hybridoma method is a widely used technique for producing monoclonal antibodies, which are identical antibodies produced of a single type of immune cell. So here's how they are created. Step 1 is mice or other small suitable animals are immunised with an antigen of interest, and the antigen is typically a protein or other molecule that triggers the immune response. Step 2, after a suitable period, the B lymphocytes from that animal are collected, and these cells can produce the antibodies for that particular antigen. Step 3 is you fuse them with these myeloma cells, which are a type of tumour cell that can rapidly divide. So that means by fusing them together and creating these hybridoma cells, we have cells that can rapidly divide but also produce the antibody of interest. So the fused cells are then cultured in a selective medium that supports the growth of those hybridomas, and the medium usually contains substances that prevents the growth of unfused myeloma cells. The hybridoma cells are screened to identify those producing antibodies specific to the antigen, and this can be done using the ELISA test, which we're going to look at in the next slide. Finally, those positive hybridomas, which means the ones that are producing the antibodies of interest, are isolated and cloned. And then lastly, you let them produce lots of those antibodies, which are harvested and can be used in different types of medicines and treatments. So the different things that they can be used for are medical treatment, medical diagnosis, pregnancy tests, and other drug tests as well. So, for example, direct monoclonal antibody therapy. Some cancers can be treated using monoclonal antibodies, which are designed with a binding site complementary in shape to the antigens on the outside of the cancer cells. The antibodies are given to cancer patients and attached to the cancer cells. While the antibodies are bound to the cancer cells, this prevents chemicals from binding to the cancer cells, which enables uncontrolled cell division. Therefore, the monoclonal antibodies prevent the cancer cells from growing, and as they are designed to only attach to cancer cells, they won't affect other normal healthy body cells. Indirect monoclonal antibody therapy is this example here. So cancer cells can also be treated with monoclonal antibodies complementary in shape to antigens on the outside of cancer cells, which have drugs attached to them. These cancer drugs are therefore delivered directly to the cancer cells and will kill them. And this reduces the harmful side effects that traditional chemotherapy and radiotherapy can produce. This is often referred to as bullet drugs, and that is because it delivers the medicine directly to the site that they're needed. So the other things that we talked about were it could be used in pregnancy tests and for diagnosis. So it has been used to diagnose influenza, hepatitis, chlamydia, prostate cancer, and even more recently in those COVID-19 tests as well. And this is all working via an ELISA test. So this is how we do our ELISA test, the enzyme-linked immunoabsorbent assay, that's what it stands for. You could use two antibodies. So first you'd have a mobile antibody, complementary to an antigen being tested for, and this has a coloured dye attached to it. You then have a second antibody, complementary in shape to the antigen is immobilised in the test. And then a third antibody is immobilised and is complementary in shape to the first one. So what you would then do is add the test sample from a patient who you want to diagnose to see if they have a particular condition to the base of a beaker. You'd wash to remove any unbound test sample. You then add the antibody complementary in shape to the antigen you are testing the presence of in the test sample. Wash to remove any unbound antibody, and then add a second antibody that is complementary in shape to the first antibody and will bind to the first one. The second antibody has the enzyme attached to it, and the substrate for the enzyme, which is colourless, is then added, and this substrate produces coloured products only in the presence of that enzyme. So the presence of the colour indicates the presence of the antigen, because you'll only have that enzyme present to cause that colour change if you had the antigen in the test sample in the first place. Then we have a look at types of immunity, and we have passive and active. We've mentioned active already, so it involves the exposure to a particular pathogen or antigen. So you make the blood cells yourself, your own memory B cells, and you're making your own antibodies. Passive immunity is when you aren't making any of these cells or antibodies yourself, you're being directly injected with the antibody. So the pathogen has not entered your body. It doesn't provide any long-term immunity, but you do have some temporary immunity because you directly receive a source of antibodies. So, for example, antibodies can be passed to a fetus through the placenta or through breast milk to a baby. That's an example of passive immunity. There's two types of active immunity. Natural, which is when you become immune because you've been exposed to the pathogen, or artificial immunity, which is when you become immune because you've received a vaccine which either had a weakened version of the pathogen or the antigens. Vaccines play a key role in immunisation and disease prevention. So they can induce passive or active immunity depending on whether you're injecting in the antibodies or the antigen. Passive immunity, as we said, is you're just directly injecting in the antibodies. But artificial active immunity is when you're being injected with small amounts of weakened or attenuated pathogen, and that will trigger a primary immune response, but with very, very limited symptoms compared to what the actual pathogen would cause you if you were infected. So therefore, if you do get reinfected with the same pathogen, you will rapidly produce antibodies and in large quantities because you would then be experiencing a secondary immune response. And this is how vaccines provide this active artificial immunity. So let's just have a look at that. We've got your primary and your secondary immune response. Now, they're not always effective in the long term, and that is because pathogens' DNA can mutate, or RNA if they have RNA. And if the genetic material mutates, it could result in a mutation that now has a bacteria or virus as a different shaped antigen on its surface. And if that's the case, the memory cells that you have circulating in your blood after your primary immune response to that vaccine will now be producing plasma cells and antibodies that are incorrect in shape for this newly shaped antigen. And this is known as antigen variability. So the last little bit then linked to vaccines is how they can protect an entire population. So an epidemic is when a disease spreads rapidly on a national level, so within one country, whereas a pandemic is when a disease spreads rapidly on a global level, like COVID-19. Mass vaccination programs prevent the further spread of the pathogen causing the disease. So these vaccines are frequently updated, resulting in booster vaccines to account for antigen variability. Again, much like COVID-19, there wasn't just one vaccine, there are multiple vaccines, and you're encouraged to take booster vaccines, especially if you're someone with a weakened immune response. And if a large enough proportion of a population are vaccinated, then herd immunity arises. And this is the concept where if most of the population are immune, that means they can't get the symptoms of the disease and they're not able to transmit the disease or transmit the pathogen which causes the disease. So if enough of the population are vaccinated and immune, then that means transmission should be so low that people that are too vulnerable to have the vaccine are protected. And you might be vulnerable because you're pregnant, you're elderly, you're very, very young, or you have a weakened immune system. So herd immunity is really important to protect the individuals who are too vulnerable to have the vaccine themselves.